Juliano, are you on a Mac? You or Mateus have a Mac? Or Lorenzo, do you have a Mac?
Yeah, I'm working on a Mac.
Okay, could I ask you to go into the folder called Hackathon Single Slides?
Yep, give me a sec. Hackathon single slides. Have to find it.
Let's see where to go. Somebody may have already replaced it.
Course slides. Ah, no, hackathon single slides.
Oh, wait a sec. Maybe somebody just deleted it. Let's see. Somebody. Okay. So there's a file there called Hodgkinson Hackathon Apoptosis and Development. And I think it may have gotten clobbered, so I have to go into Google Docs and it's a key file, .key.
Yeah, I opened it yesterday. I need to work on the
Can you
previous.
save it as a PDF?
I will try, but I have to access the folder at the moment. So, Hodgkinson hackathon apoptosis. This one.
Yes, I could share it with you.
Roll of zebrafish, etc.
Right, because I can't open that file.
Even if you put the eye on the slide directly on Google Drive, you click the eye.
You mean try to open it in slides?
Directly in Google Drive.
Well, my problem is I need to import it into PowerPoint.
I see.
And I can't open it. I can't open it as anything. I mean, I could try to do a cloud convert, but I don't. It's not a format that I can use.
I'm downloading it now. It's a keynote, so
Right.
I have it on my PC, so I can export to PDF right now. Give me a second that it opens. Save as, export to PDF. PDF is fine?
PDF or yes, that would be fine because I can open a
PowerPoint.
PDF as a PowerPoint.
Here you go. You should have them when they send.
Thank you. Thank you very much.
No problems.
Did you email it to me or did you put it back in the folder?
Email. I replied to the mail. It's just sent now.
Fine. Okay, thank you.
What shall I do with the videos?
Oh, there were videos in that?
No. On those that I recorded yesterday, shall I upload?
Oh, yes, to the student materials videos folders.
Yeah. The only problem is that I cannot include the closed caption. I don't have an easy way to do that. I have the file, but I don't have an app that can blend this.
You don't have to, just put them as a separate file.
Okay, perfect.
That would
So
be fine. Okay, so let me get that last slide deck pulled in. Okay, I still don't have it. Huh, there it is.
okay.
Oh, you were able to export it as a PowerPoint. That's great.
Both PDF or PowerPoint, whatever you like better.
Although it looks like the doesn't have to be perfect. It just has to be good enough. Oops. All right. Huh. Okay. Can I ask the other co-hosts to do a local record, please? That's especially important if you're giving the talk. I'll wait another minute or two for people to join in, and then we'll get started. Okay, I'd like to welcome everybody back to the second day of our CompuCell workshop. I hope people were able to get some rest and process all the material that was presented yesterday. We're going to have a lot of additional material today. I hope it'll be interesting for people. As always, I need to remind people that the series is being live streamed and recorded, so please keep that in mind. I also have had some issues with being kicked out of Zoom. There was a Zoom update last night that seems to be causing some problems, so please be patient if there are issues of that kind. Today, we're going to start out with a talk by Juliano, who's going to talk to us about how to apply forces to cells, move cells around, and collective motion in cells. We're then going to briefly review the hackathon lightning ideas and have a presentation on some COVID modeling that was done by TJ Segal. And then in the afternoon, we're going to start on how to build models of chemical fields and gradients. And that's going to be taught by Josua Aponte-Serrano. So that's the agenda for today. If you have ideas for the hackathon that you didn't put into a slide deck, that's fine. Please speak up at the end of the lightning presentations, and I will do some reorganizing of the hackathon slides during the next hour. Again, the schedule will be more or less the same. We'll start our official meeting at 10:30. We will have a short break. Then we'll have the hackathon lightning talks, the discussion of the hackathon, and TJ's presentation of COVID models. We'll have a slightly longer break to catch a bite to eat. And then in the late afternoon, we'll have Josh doing the presentation of chemical field modeling. And we'll conclude with question session if people need that. I've tried to put together the lightning talks and material that were presented in the decks. It's fine if you have a hackathon idea that comes up after today that wasn't included in the deck. That's also, if you want to present something today verbally, that's fine too. This is the list of people who've submitted material. If you could be ready to present, this should be the order, and I'll put it also in the chat. Havid Azimi, AJ Chitness, David Fagbemi, Farnoosh Farapur, Hamad Fatuyimbo, Megan Hassa, Yajun He, Aaron Hodgkinson, Yana Shuhas, Iman Kafianatari, Maria Raffo, Lorenzo Veschini, and somebody presented a very interesting slide on invasion and breast cancer, but they didn't put their name on it. So if you own that slide and you can tell me who you are, I can add your name. Otherwise, when we get to that slide, please speak up. And if you've put in the slide and you decide you don't want to do it, that's all right too. Just let us know. But that's the planned order of discussion. As always, people were using the Zoom quite effectively yesterday. If things are too slow, too fast, you need help, please feel free to use the Zoom buttons. We've been trying to put the recorded videos of the Zoom sessions up online. Not all of them are there yet, but we're doing our best to get all of them up. The slide deck should be available for people. Any suggestions you can make to us to be more effective would be appreciated. And again, the hackathon will be on Saturday and Sunday of next week, well, this week, say next week, this week, and I hope that people will participate. We have the usual NanoHub links, and I hope people are having NanoHub work better. I know there were some issues yesterday during my talk when people were getting started. Some people have found some bugs, so those have been reported and we'll work on those as well. I hope people who decide to use the desktop app all have that working now too. I know there were some issues with people before. And there's the usual resource page. I especially draw people's attention to the Tellurium manuals and videos that are available because we're going to start tomorrow afternoon with a very brief introduction to using Tellurium within CompuCell. And so those could be helpful. Again, I really encourage people to print out those quick reference guides and use those. They're very convenient to have printed out so that you don't have to keep switching back and forth when you go from programming to the screen. That really is about all I had for today. We have the Slack channel. As anybody have, I hope everybody's been able to get into that. We seem to have been using the chat more than the Slack yesterday. That's fine too. And I think the key thing at this point is to see if there are any other points of discussion or questions that people have for the people who spoke yesterday or concerns about today, suggestions that you'd like to bring up. Again, I'd like to thank everybody who's involved in the meeting for their time and effort. So that's really all I had for the introduction for today. I'd like to open it up for conversation and questions. If not, you're free to go off and come back at 10:30. But I wanted to give everybody the opportunity to weigh in on how we can do things better, what's working, what's not working, anything that you would like to contribute.
So there is a question in chat about downloading from NanoHub. There is a way, and it's pretty much the reverse of what we did yesterday to upload. So you need to open Jupyter Notebooks, and you're going to be presented with that page of the directory of your files. And in there, you can select what files you want to download and so on.
You have time to demonstrate it if you want. Juliano, we have.
Okay, let me see if I can do it live. If I don't have to eat my words. Hub is being slow to open.
Can somebody turn on live caption?
Um, I think I activated, but I'm not sure if I activated it for me or for everybody.
The
live
captions are turned on.
Yep. Yeah, you need to go to the more menu on Zoom, and then there is something about subtitles.
But they are enabled.
Well, I guess we'll wait for 15 minutes until NanoHub doesn't open Jupyter. Yes, that would work, Cicely. Already have it open. Okay. Yeah, so the thing is, I think you can't do a whole folder. You have to do it file by file. There you go.
Oh, I see. Okay,
Yeah.
perfect. Right.
Yeah,
Okay,
it's
that's brilliant.
kind of annoying because you have to do it file by file, but
And so if you've got your simulation, can you just, as long as you put it back in a folder that's called simulation and all that stuff, then
yes,
will it
yes,
still
yeah,
run?
Okay. Yeah, as long as you keep the folder structure, it should work.
Perfect.
That's
Thank
why
you very
we,
much.
that's why we usually zip
Yeah.
things. So in Twit,
Yeah.
you can zip your file, and then that will
Okay,
create
yeah.
a single file to download.
Oh, awesome. Right, brilliant. That's great.
Yeah.
Thank you very
So
much.
there's a Twit button that says to zip it, and that will save the files as a zip.
Yep.
And when you unpack it, you'll notice that it'll give a warning about a non-unique file name, but it'll work.
Okay, that's brilliant. Thank you very much.
No worries. And at least I think there should be an easier way to upload and download stuff from NanoHub. So maybe if we all start sending them tickets requesting that functionality, maybe we'll get there.
Any other questions? I feel a little guilty having everybody sitting waiting. You're welcome to go get a cup of tea. I put the discussion order for the. I'll reshare the screen for the discussion order for the hackathon talks. Again, I remind you that if you didn't share a slide, that's fine. You can speak up and introduce yourself after the list. If you would like to join somebody who has presented, you should have their contact information, and please feel free to join up together to work on projects. But this is the list of people we have so far.
Imam, yes, you can. I've done it before. But CompuCell also can save the cell lattice as a VTK file. And the VTK file has, if I'm not mistaken, all the info from all of the cells. And if I'm wrong, please, TJ or Maciek, if he's here, correct me.
Sure. Yeah, that's correct. So at whatever steps, or really at a frequency that you specify, you can write the lattice values as well as associated data with the lattice. So if you have a certain number of cell types specified and things like that.
Yep, and the chemical field gradient.
Field values. Yep. Field values and names, name of each field.
Yeah. But you could also create a NumPy matrix and do it by hand inside Python. I've done it before. It's not the fastest thing to do, go over every pixel of the simulation, but it's doable.
So there was a question about the hackathon lightning talks. The plan is that I have your slides so that instead of having to reshare the screen between people, which takes a lot of time, I have the deck with all the slides in it, I hope. If they're missing, then we'll have to come back and fix it. But what we'll do is I'll put up the deck. I'll show the first slide. I'll say, AJ, would you please explain your idea? And then you'll explain your idea. And then I'll show the next slide and say, David, would you please explain your idea? I hope that works. It's a little more efficient, I think, than having to swap the screen shares. There's always some risk of of losing the Zoom connection when we do that. And again, once we've gone through all the slides I have, if there are people who have ideas or slides that they want to present that weren't included, you're free to speak up and we can talk about it. And I'll try in the next day or so to be able to talk to people about trying to refine their project ideas a little bit. I encourage people to reach out to the instructors so that we don't overload any single instructor, and then discuss their project ideas one-on-one to help refine them a little bit before we start. And I'll also try to go in and put in the extra information that people had about contact information and expertise that you're looking for your slides. If I can do that between now and the start of the next session. TJ, there was a question about exporting 3D simulation shapes as objects. Could you want to take a stab at answering that?
Sure. So by 3D simulation shape, would this be something like exporting the 3D data of where cells are? Something to that effect? Okay, yes. So we were discussing a few minutes ago about exporting to VTK. So that's the standard way of exporting lattice data, whether the lattice be 2D in effect or 3D. I say 2D in effect, and the two-dimensional simulations that we do are in fact they have the same structure as the three-dimensional simulations. They just have one voxel along a particular direction. And then, so Jim pointed out that exporting to STL might be a good hackathon topic, and we do have a module reserved for sort of practical everyday CC3D, and yeah, this might be a good activity to cover in that content.
Okay, Juliano, do you want to queue up your splash slide for your talk? There's a question from Polly. Go ahead.
Okay. It's 10:30. Don't know if we should get started or wait a few more minutes.
Why don't we wait till 10:03?
Okay.
That seems to be the canonical time.
Okay, it's 10:33, so let's get started. Hello again, everybody. Again, my name is Juliano. I should remind you all that we are both being recorded and live streamed to YouTube. And in this lecture, I'm gonna show you how to apply forces to the cells and how we can use those forces to make them move as a group. And once again, and also show the relationship of force and velocity, that it's not force and acceleration that are linearly related, but force and velocity, and that there's also a speed limit in cellular POTS models. Okay, and I also will show how to visualize more things with extra fields. So what we will do today is a simple simulation of wound healing. In reality, as the figure there shows, it's a complicated process in 3D with several cell types, coordinating their behaviors, and so on until you get a scar. The simulation we will do is way simpler. It's going to be a scratch assay simulation, which is 2D, which already helps, and it only has a couple of cell types. In our case, we're only going to have one cell type. Move. So how we make the cells move is by applying forces, as I spoiled already. If you don't apply forces to cells or have them keep them attacks, they're only gonna wiggle around the position they are at. And the way to apply forces is by using the external potential plugin, and it defines a directional energy. So if you think about the potential energy of gravity acting on you right now, that is a directional energy pointing downwards. But for cells, this force that we are applying really represents acting polarization or information that the cells are passing to each other, trying to coordinate the movement. So first, we need to start a simulation. So I already have CompuCell open here on NanoHub. And again, I'm going to open Twedit with the pen and paper icon, or here, start Twedit. Make that bigger. Let me resize this. And again, we are starting a new project. So CC3 project, new CC3 project. We need to select where we are going to save it. Again, I'm going to make a new folder here. Choose, and I'm going to name my simulation external potential collective motion. And I hit next here. And in this case, we are going to leave almost everything here by default. Now we are going to use the no flux boundary conditions. I want to show you something different. We'll change the copy range to 2. That's just a personal preference, but I think it works best. And we are going to leave the rectangular slab selected. After all, if we look here, the cells start in 2 rectangular slabs and then move to the middle. Oh, and by the way, if there is some noise on the background, I'm sorry, they are doing landscaping outside. Okay, so that's all for the general simulation properties. Now we are gonna have 2 cell types. One that is actually a cell. I'm gonna call it MOV for moving. And the other one is gonna be a wall cell that we are gonna build around the simulation. And since it's a wall, it shouldn't move. So we select freeze there. And if you haven't selected freeze there, you can also just select and unselect it here. And with this wall cell, we are gonna build a 1 pixel deep wall around the simulation domain. And the reason for that is so that we can actually define a contact energy with the edge of the simulation. If we have nothing there, the contact energy with nothing is 0. And so our cells will be quite sticky to the walls, and it will be hard to deal with that. But by building around a wall around that is a cell, it's easier to control. Okay, next, again, we are not going to use any chemical fields. That's going to come later on Josh's lecture. And for properties, we are going to have contact as usual, volume local flex. We already have center of mass selected. And here I have cell neighbors, but this is actually a bit outdated when we don't need cell neighbors. And that's it. So if I go back to the start, select where you want the simulation to live, give it a name, defaults here, to here, rectangular slab, moving cell, wall that is frozen, no chemicals, contact, volume local flex, and center of mass. So the difference between volume flex and volume local flex is that volume flex defines the volume constraint on a cell type basis, and the local flex defines the cell volume on a cell basis. So we can have a bunch of cells of the same type with different target volumes. And the pixel copy range 2 is not the neighbor order in the contact energy plugin. It's really unfortunate that both those things have the same name in CompuCell. And again, that's all. So once more, name and place, defaults, rectangular, tube, mauve and wall with wall frozen, no chemicals, contact, volume, local, and center of mass. And that's it. Okay, we have our simulation ready. And one of the things is, well, let me just show you. Since we are using volume local flex, oh, I need to open the correct simulation. Since we are using volume local flex, our cells don't start with any volume constraint in actually set. So if I just hit play here, the cells are behaving as foams were, but with some wall cells thrown in to the mix, which we definitely do not want. And here, if we go to the XML, let me show you what's different. The thing that is different here is, well, the wall cell has the freeze here in its tags. And we have, wait, did I? Sorry, I forgot to add it here on the slide. There was one more plugin which is really important for us that I forgot about, but it's fine because to edit can come to the help. So we forgot a plugin, which is the external potential plugin, which is the main point of this lecture. So if we come to the XML and place our cursor anywhere outside of opening and closing tags for plugins and steppables, we come here to CC3D ML, which is the same as the CC3D Python menu, but for the XML. And go to plugins, we have here somewhere external potential. So we just paste that in. And we see here one option of the plugin is pixel-based. We could also use center of mass-based. That will change how CompuCell computes the energies internally. With pixel-based, any pixel copy attempt in the correct direction will diminish the energy. With center of mass, the changing pixel would need to change the center of mass to the correct direction. And we also see here that it also pasted parameters already for the mauve and wall cells, but we are not going to use these because if we define these energies here, we cannot change them easily inside of our steppable, which is where we are going to do the calculations. So either comment out these lines, and I think you noticed yesterday that XML comments are not pretty. So the easiest thing to do is just select both lines. And since the shortcut is not working right inside NanoHub, we come here to edit and click the block comment / uncomment. Now the text turned green as the other comment, so we know that worked. And the other thing I wanted to show is that here we have a bunch of wall cells in the middle. And that's because all the way down on the uniform initializer, we have mauve and wall inside types. I commented out, I didn't uncomment. So what I commented out were these extra lines on the external potential. So again, I forgot about the external potential, which is silly, but to edit, we go here to CC3DML plugins and click on the external potential. And if I click again, I get a warning that it's already loaded. And what we want to remove are the two lines that start with external potential parameters. Because otherwise it's going to be hard for us to actually change these applied energies. So either comment those out or straight up just remove the lines. Both of those are fine options. And then down here on the uniform initializer, we only want to have moving cells. So we remove wall from the types.
Excuse me, you select the external potential or external potential local flex in the plugins.
Is there a local flex?
Because in the wizard panel, we have two options there.
Oh, the wizard. Select the local flex. Yep. Yeah. If you select the other one, it would add these lines. So the difference between those two in the wizard is that if you select the local flex, it won't add these lines, which automatically tell CompuCell that you're gonna define stuff on a cell basis. And with these lines, you're telling CompuCell that you are defining it on a type basis. But if you selected the wrong one, you can just do what I did here of commenting those out or removing the lines. And in the uniform initializer, we removed the white wall type from inside the types tag. So if I rerun this, now there are no wall types. But the cells still disappear because we actually didn't tell them that they shouldn't. And computers are very literal. So before we actually keep our cells alive, we are gonna build that wall. And that's very simple to do. We want a 1 pixel deep wall around the whole lattice. So we come to the steppables file, and in the start function, we ask to edit how to do it. So we go to CC3D Python, and this is in simulation. And sometimes it's hard to remember where things are, but if you patiently go through, you'll probably miss what you're looking for a couple of times and then find it. And in simulation, we hit build wall. And here's the command, which is aptly named build wall. And we just need to pass the cell type we want the wall to be. So again, coming to the steppables file and CC3D Python simulation build wall. And we called our wall type wall. So we just replace that by wall all capitalized. So when we are referencing cell types in the steppable, the type name has to be capitalized. Now,
if it is like the combination of the capital and not capital letters in the cell type, so we have to write all them in capital letter, right?
Yes, it doesn't matter if I name them mauve like this, or mauve, or mauve, or mauve, or mauve. In mauve, V at the end for moving. It's going to be all capital inside the Python.
Okay, thanks.
And yeah, I could have called them moving, but mauve is less typing. Yes, they do need to be consistent within the XML. That's another thing. That's why I put it back as it were, because otherwise it would complain down here in the contact and in the uniform initializer. So now I've built a wall. And if I hit this again and wiggle, we can see that some yellow showed up at the edges. And since it's 1 pixel, it's kind of hard to see. But if we zoom in, we can see the wall there in green. And now, yet again, before we keep our cells alive, we don't want them to be in a square in the middle. We actually want 2 rectangles at the edges, right? So how to do that? Well, here back in the XML, in the uniform initializer, you can notice that there is a region tag here. And there's nothing stopping us from copying the region tag from the beginning to end and pasting it again. And now we can define 2 regions. So the first one, it's going to be the left side rectangle. So we want it to start at the beginning for X. I'm going to align my rectangles vertically with a gap in the middle. So X from 0 to, seems I selected 70 before, so I'm going to take my word as being good. And Y, yes. So in here I used 1 and not 0 because we have a wall in the first pixel. So it should be 1 instead of 0 there. And the Y should be 55 instead of 56 because of the wall as well. Although I don't think it would be too much of an issue in this case because the creation of cells by the uniform initializer happens before our creation of the wall. So the wall would just squish some cells, which not very good for the cells, but okay for us. Yeah, probably should. I'm gonna be honest, I'm never too sure how the index happens in this boxes here on the step rules, but I know that with 1 to 255 works. But no, you're right in being picky. Yeah, the Z has to be 0 and 1, even though there is only one Z. It's weird like that. And the second region, well, we wanted to start after 70. So let's give a gap of 100 pixels, start that at 170 and go to 255. And Y should be the whole Y direction. So it should be as before, 1 and 255. And the final thing is I prefer cells that are 8 by 8 instead of 7 by 7. So I'm just gonna change the width to be 8 here. If you don't see your wall, even after zooming in, what may have happened is that you forgot to set it to be frozen. And in that case, the wall is gonna vanish immediately. So to make sure that your wall cell is frozen, you go back to the cell type plugin in the XML and check that there is this freeze equals open close apostrophes. Well, in that case, did you use the build wall with the correct cell type? If those two things are right, I'm gonna ask for someone to take over. Okay, so now we have our regions, and let's make sure that they are what we want. Okay, we go. There we go. Looks right. There is still the wall there. Some cells got squished because of how things ended up happening, which is somewhat unfortunate, but it's okay. To pause at the initial condition, what I'm doing, instead of hitting play or run, which would just, start running, I use the step next to the play. And that will do a single step, so we can just observe the initial condition. Okay, so now we can finally keep our cells alive. So what we do is we are gonna add a loop at the start after we build a wall over all cells of type moving. Stephen, wall should be all capitalized. And to visit all cells of a type, we ask to edit how. So C3D Python, we're visiting something, so visit, and all cells of given type. And here we go. And as the pasted code suggests, we can do as many types as we want in this loop at the same time. In our case, we're only going to go over the moving cells. So I'm going to remove everything here and put in mov all capital. And finally, I'm going to remove the print here. I don't want to print anything to the console. Printing stuff is actually quite slow for whatever reason to console. It's actually slower than writing to a file, or as slow as, which is weird. And we want to set their target volume and lambda volume. And again, we don't need to remember the names there. We just ask to edit for it. So C3D Python cell constraints, as we are constraining their volume, and change target volume. There it is. We don't want 25, we want 64. As we started the cells with a width of 8. And again, cell Python, C3D Python cell constraints, and change lambda volume. Has 2 here. I prefer 8. I think things work just a little bit better with 8 instead of 2. And I'm just going to remove some extra spaces that we don't need here. And now the cells should stay alive and just wiggle around. So lambda volume is, so the way that the volume constraint and the surface constraint work is by kind of like springs. So the cell, if it deviates from its target volume, it's going to try to go back to it, as a string would try to go back to its relaxed position. Realize that I may be going a bit fast. So, and the lambda for volume and the other parameters as well are like the K constant of the spring. So target volume is kind of self-explanatory. It's the volume that the cells will try to be at, and they try to be at that volume as if they were springs, but a volumetric spring. And the lambda volume is the strength of the spring. And how I did all of this was first for the build wall to a CC3 Python simulation build wall and passing the wall type all caps, which is here. And then this loop, again, I asked to edit how to do it by going to CC3 Python visit, since we are visiting cells, and all cells of a given type. And it's gonna paste some more stuff here inside of the parentheses, but we are only going through the moving cells, which I called MOV. So I just pass the cell type MOV all caps. And then on the regions, what I did was I copied the region that was already there down here. And I changed the X start and X end for both of them, so that there are two rectangles with a with a gap in between them. And I also told it that it should only, those rectangles should only contain the moving cells. I have I'm using the same numbers as here on my presentation. So the first region is from 1 to 70 with the whole of Y. And the second region is from 170 to 255 with the whole of Y. Target volume is here. So I set 64 because we are starting our cells with a 8 width. So, and to actually, you don't need to copy from here. you may make a typo. What you do is go to CC3D Python and ask to edit about the cell constraints. So you want the target volume, not surface, and lambda volume.
When we use self-built wall, does it automatically build the wall all over the lattice, right? So if
Yes.
we want to make on a specific parts, I mean the wall, then we need to define it in the region part, or
Yes, you could use the region, but if I have time at the end, I can show you how to be more precise with your walls. If I spoil my presentation and go all the way to the end, here I built kind of like swimming lanes for cells, and the lanes are walls. So I may show how to do a pattern like this.
yeah, that would be useful. Thank you.
The wound won't close yet, as we are not actually telling the cells to do anything. They're just wiggling about. We are not pushing them around. So they are just being there, chilling. Feeding to experimental data is a constant battle. We try our hardest to do it, but sometimes doesn't work. But things like the target volume, this could really be arbitrary, because a pixel is a pixel. It has no real units. If I say that my pixel represent 1 micrometer, I'm saying that my cells are 8 by 8 micrometers. But I could say that 1 pixel is half a micrometer, and in that case, my cells would be 4 by 4 micrometers. And I could say that my pixel is a meter, and I would have the hugest cell ever. So the next thing that we need to do is actually to, well, select where the cells are going to move and how, at least the initial direction for their movement. And I'm sure when a wound happens, there's a ton of signaling in between cells that actually detects where the wound is. And they actually start coordinating from the start. But a easy way or straightforward way to select the initial direction is to just select a random direction. That's perfectly reasonable. As I start, you could afterwards change your mind and do something else. So what I'm going to use is NumPy's random number generators. And so I'm going to come to the start of the stepable file, and I'm going to first import NumPy as NP. And this would already give me access to all of the random number generators from NumPy. and they would be accessed something like np.random.uniform, which is one of the generator types. But that's quite a bit of typing. So instead, what I usually do is I import those separately by doing from NumPy import random as RNG for random number generator. So let me just, I can copy from inside NanoHub. And this way we can select the initial direction that the cell will try to move towards. And we are going to call that direction the bias, because it's not the direction that the cell will move towards, but the direction that it is biased to move towards. So we need, well, a vector of 2 components, and we could generate 2 numbers and make sure that the resulting vector is unitary, or we generate only 1 number and get 2 directions for the vector from that number, which of course is generating a angle. So here in the loop over the cells, since we are going to give each of the cell a different initial direction bias. So I'm going to have theta equals to RNG and G, not M, uniform. And I want the whole circle as a possibility. So anywhere in between 0 and 2 pi. And pi lives inside of NumPy, and you can access it by doing numpy.pi. Then I can now define my bias, which I'm gonna, there on my presentation, I gave it a long name. Here, I'm just gonna call it bias. And this is going to be a NumPy array, which is similar to lists and tuples, but it makes doing operations to them way easier than with lists or tuples. So it's going to be a NumPy array, so NP array. And to this NumPy array, we have to actually pass either a list or a tuple that will be converted into an array. If we just do the numbers that we want, say 1, 2, 3, I'm not even sure what would happen. Maybe it would throw an error, but it definitely wouldn't be what we want to do. Yeah. So it threw an error. So it's NP array, open parentheses again inside of it. And then the X direction is going to be the cosine of that angle. So NumPy cosine of theta. And NumPy sine of theta. And since we are in 2D, that would be enough. But just in case we want to later on change this to 3D, I'm already going to add a third element here, but I'm going to set it to 0. Now I have to close the tuple inside of the array. So for this part, what I did was first I imported NumPy as NP. And then from NumPy, I imported random separately as RNG for random number generators. In Python, there is no difference between double and single quotes.
You have to use them in pairs. So
Yes.
if you use a single quote at the beginning, you have to use a single quote at the end.
Yep.
And Python also has a special thing, which is a triple quote, which identifies text with line breaks in it.
Yep. And then the next thing I did was I rolling a 2-pi-sided dice to define a angle for the initial direction. And I'm saving that to theta. And then to get the direction, I'm getting the cosine and sine of theta. I have those inside of a tuple that I'm passing into this function NumPy array that will actually turn it into a vector. So for the iterating over cells, just remember that you're visiting cells, and that will make it easier to remember where it is, but it is in CC3 Python visit for visiting stuff, and all cells of given type. And that will paste this with more stuff inside this parentheses here, but we only want the moving cells. So I'm passing self.mov there.
Sorry, why did you use two parentheses in the array, NumPy array?
Yeah, because the NumPy array is expecting to be given either a list or a tuple or something similar. If we just had cosine, sine, and 0, we would get this error. I have this simulation, by the way, zipped here in its final form. And in here, I put in a bunch of links to something. Yeah, this line. Recent projects. And in this zip here, I put in a bunch of links to the NumPy functions that I'm using. So let me just copy. Nothing. Where is it? Here. So if you want to read more about NumPy arrays, there's the documentation. Let me go back here. Yep, Iman's explanation is 100% right. And this is all well and good, but what we are doing here is we are creating a random number, creating a vector, and then throwing those out. We can make cells remember information that is relevant to them by using cell dictionaries. So cell dictionaries are a regular Python dictionary. So you can put anything inside of cell dictionaries, even other dictionaries, as long as you give each element a different key. So we are going to tell ourselves to remember their bias and remember their angle by doing cell dict. And this is one thing that I'm not sure if NumPy knows how. Might be the first one that we come across today. So for it to remember its bias, I'm going to add a key to the cell dict called bias and set that to be the bias. And I'm going to do the same thing for the angle. So now each cell remembers their bias and their angle. They are still not doing anything with that information, but they know it. No, they are not reserved properties. This dictionary starts empty, and as it is a dictionary, you are free to do anything. And this bias and angle are things that we are defining for our simulation that we need. There is a Reddit knows how to do cell dictionaries. So it's CC3 Python cell attributes access modified dictionary attribute.
And maybe it's worth emphasizing that the built-in cell dictionaries are one of the most powerful things that you've got in CompuCell.
Yes, they
because
are.
It means you could attach any kind of attribute. For example, if you wanted to keep track of the lineage of a cell, if you were doing a cancer simulation, and so you wanted to build a list of the ancestor of the cell, the ancestor of that, the ancestor of that, you could put that as a dictionary entry.
Yep.
If you want to calculate the previous coordinates of the cell and in time to calculate the history of the velocity of the cell, you can do that as a dictionary entry. You'll also find that we use dictionaries to communicate when we want to connect subcellular models written in SPML, MABOS, or antimony with CompuCell. And that's also quite powerful. There's also a free-floating dictionary which belongs to the simulation as a whole. And the advantage of using that is that the information in that dictionary is available across steppables. So if you put information into that dictionary, it effectively globalizes it and makes it available to everything in wherever you are in the simulation.
Yep, here. We are not going to use it here, but say, say we had more than one steppable and we had one that just initializes all of our parameters, but we are only going to use them in another steppable. We could come here to CC3 Python simulation and here, shared steppable dictionary. And again, this is a dictionary, so you just give it a key and a value, and it would be accessible to the rest of the code. Yes, we are creating the bias and the angle. They don't exist until we tell them to exist. And now we are finally gonna apply this bias to do something, because as I said, the cells know that information, but okay, what do I do with this? And we are going to use the directions of the bias to apply the force that we want. So still in the same loop, we are gonna go to CC3D Python, cell constraints, and apply force to cell. So again, CC3D Python, cell constraints, first one, apply force to cell. And then already pasted some numbers here for X and Y that we're not going to use. What we will do is, well, we're gonna leave the Z as 0. After all, we are in 2D. And then for X and Y, lambda vec X, lambda vec Y, we're gonna change the number to -20. And it's weird to be using a negative number, I know, but wait for the next slide. And the first one is X, so we want the X direction of bias. So we multiply that 20 there by the value that we've stored on the cell dict. And this element of the value of the key bias inside of the cell dictionary is a vector, and X it is the first element of the vector. So we do cell dict bias, and then inside a pair of square brackets, 0. And same idea for Y, but instead of 0, 1. And now the cells are actually doing something besides chilling with their brethren. So if I go back here and hit play now, and we go. Now we can see that the cells are separating and doing weird stuff, but we are only told them where to go at the beginning. And we are not updating the bias. So okay, I have to go in this direction. I'm gonna go in this direction. And that's what they are doing. So we actually need a way to update the bias. I could have done an under step, but it's best practice to initialize stuff at the beginning, so at the start. And if I had just used the loop on the step for every time step, it would set the target volume, set the lambda volume, create a theta, and the update to bias would be random. So I would have to put this loop inside a if statement in the step function. So it's just easier and better practice to do it inside the start function.
Maybe we need to apply the forces inside the simulation, like after 1000 Monte Carlo step. So can we have access to the dictionaries And once we defined it in the step function, and I mean the start function, then we use them in this step.
Yes, yes. The cell carries its dictionary around wherever it goes, and it remembers its dictionary wherever it is. So this is also an easy way to set parameters in one stepable and use them in another, because the cells remember.
Oh, you mean that we don't need to define the steppable variable that you said before in order to have access in the cell dictionaries in the other steppables?
Yeah, no, each cell remembers the whole of its bias, its dictionary everywhere.
Okay.
Yep. And
TJ made a good point in the chat, which is one thing you do want to be careful about when you're adding things to dictionaries is that you don't want to add complex objects like cells to a dictionary. It's okay to add a cell ID when we get to attributes. Cells, each cell has a unique index, cell.id, which tells you the number of the cell. It's okay to add the cell to the dictionary, a cell ID to the dictionary, but you don't want to add a cell object to a dictionary. That can cause problems.
Yep. And yes, Farnoosh, that's exactly the reason, to make accessing and changing this data easier. It's 0 in X because, well, bias is a tree length vector, and the first element of the vector is the X direction, the X length of the vector, and the second is the Y length of the vector. And Python is 0 indexed. So the first is 0 instead of 1, which is a common thing in computer science. And even though I deal with it for over 10 years now, I still don't like it. The reason for the negative here is because we are defining energies in CompuCell pretty much always. And the relationship between energies and forces is that force is the negative gradient of the energy. So if we say this, instead of bias 0, we just had -20 here and -20 here, it would mean that the cell would move in from down here, 0, 0, all the way to the top here. But if we had 20, 20, it would be the reverse, from max X and Y to 0. That's just a weirdness about this plugin. Okay, now we can actually start coordinating the cell movement. Cell movement coordination is very complicated. There are intercell communications, signaling, trying to align their actin fibers, and so on. But the good thing about computer models is that we can simplify stuff until we get the behavior we want, and then recomplicate them as needed for the problem we are doing. So in our case, we are going to just do the simplification part. So the way we are going to coordinate cell movement is with this formula here. So we are going to define the next bias direction to be some percentage of the previous bias direction plus the complementary percentage of the direction that the cell moved. So the normalized velocity here. And in one thing that I thought about while doing this is that it might be interesting to have this alpha here change during simulation time. So at the beginning, we started out the bias randomly. So it would make sense to have a very small alpha so that their memory of their bias is short and they can align faster, and then increase that alpha during simulation so that it cares more about how it was going. But we are not going to do that. And how this actually aligns the bias is, well, by the cells pushing each other. So say we have two cells here, green and blue, and green cell has a bias that goes straight into blue. Well, it can't really move there. I mean, it will push the blue cell a bit, but it's going to be hard. So its movement is actually going to be somewhat to the top instead of going here. And when we update the bias, we are going to make the new bias shift a little bit towards the velocity before, and so on. And when we have however many cells are here, which is 641, then they will coordinate. They will end up coordinating. It's not without much fuss. So first thing we will do is actually calculate the cell velocity. In the past, we had to actually keep track of the previous center of mass, but nowadays, CompuCell automatically knows the current center of mass and the previous center of mass. So we are going to move to the step function, and we are going to change this loop. So and this actually happened to me. Well, let me do it and show you why it won't work. Well, it may work for a while, but it will break eventually. So if I just keep this loop over all cells, eventually things will break, because I will want to update the bias inside the cell dict of a cell that doesn't have a bias inside its cell dict. Why? Because in this loop at the start, we went through the moving cells, but we have one cell that is not moving, which is the wall. So we actually have to change this loop here to be over only the moving cells. So I'm just gonna remove it in its entirety. And I'm gonna again ask to edit how to do the visiting and go to CCG Python, visit all cells of a given type. Or I could have just, gone to my loop in the start and copied the for, which is also fine. So the first thing, actually relaunch the pool just to make sure that I'm not. Fair enough. Okay, so going over what I did at the start before continuing. With build a wall, a simple function, we just, pass it in the wall type. We are going over our moving cells. we are setting their volumes and their volume constraints, both the target and the lambda. Then I'm rolling a 2-pi-sided dice. Well, actually has way more sides than that. So a random number generator uniform is going to create any number in between 0 and 2 pi here. And then with this random angle that we've created, I'm calculating its cosine, which will give us the bias direction in X, and its sine that will give us the bias direction in Y. We have those two and 0 just in case we want to go to 3D later. Inside a tuple here, you see the parentheses here and here. And that tuple is being passed into the function numpy array, which transforms a tuple or list into a numpy vector or numpy array. And the reason for that is just that dealing with numpy arrays for doing mathematics is way easier than dealing with lists or tuples. And then we are telling the cells that they should remember their bias and their theta. We are making them remember the bias because this is how we are going to actually do the update here of the bias. Each cell has its own current bias, and each cell needs to know its next bias. So, and that information is individual to the cell. That's true. NumPy arrays are very quick. Lists are very slow. Then we're also saving the angle because, well, no real reason not to save it. And we are going to see how to visualize it nicely afterwards. Okay, so updating. Yeah. So in this step, we're going to add the velocity formula in this step. We are again going over all of the moving cells. I'm going to remove the sprint here, and I'm going to create a new cell dictionary called velocity. Remember to close my strings. And again, we are going to create a tuple that we are going to pass to a NumPy array. So we have here the array, and here the tuple will create. And in here, let me just go to the previous slide real quick. What I want is cell attributes, and I want to know its center of mass. So we know that the center of mass is, well, center of mass, COM, X, Y, and Z. So the velocity in X is going to be its center of mass in X right now, minus its center of mass in the past. And nowadays, CompuCell remembers the past center of mass, and it's the same thing, X, COM for center of mass, and then prev with capital P for previous.
How did you know that that method exists?
The previous XCOM, I found out that it exists recently, and I just remembered. But it is something that we will add here to this menu, but it's not there yet. But the center of mass is there.
The one problem that I have, I'm always searching for different methods for example, for cell, I find that there's ID, type, and YCOM. So sometimes it's difficult to find what tools we have, you know, to go through.
That's the reason we have that cheat sheet to print out. It doesn't list absolutely everything because sometimes things get added, but it has a pretty good list of what's available. So maybe somebody could share the link to the cheat sheet again. And certainly as CompuCell is updated and features are added, sometimes those takes time for them to find their way onto the cheat sheet. But the majority of those methods and attributes should be there.
Yeah, I was searching about the files, I mean the Python files, in order to find the basic files like the source code, in order to find the methods there, like steppable base file, and also cell G to find the methods. So I wanted to know which files contain those methods that I can go through them and get the updated ones when you know the cheat sheet is not updated yet, you know.
I'm adding the 0 just in case we want to transform this to 3D later on. And David, I think it's best you talk with TJ on a breakout about that stuff. So now we have part of the things that we need to actually update the bias. We have the velocity, but we actually want the normalized velocity. And that's easy to do with NumPy as well. Oops. So here, if I do the norm, that will give us the direction. I can just do cell-dict velocity. And in here, I'm going to put a column. And then I'm going to divide it by numpy dot linaug, which is NumPy's sub thing for linear algebra, and norm of cell velocity. And here the colon means for every element of this vector. So for every element of this vector, I'm dividing by the norm of the whole vector. And that's it to get the normalized velocity. And now we only need to create a new bias. So we do that by, again, copying over the cell dictionary bias. I have don't worry about the backslash. I only put it there because I broke the line. But if the whole command is in a single line, you don't need the backslash. The backslash is for multi-line comments. So the new bias, well, we need to update all of its elements. So column there. It's going to be alpha, which we haven't defined yet, times itself, plus, here I'm going to use a backslash again. Don't worry about it. Times 1 minus alpha v norm. And v norm also, we need the column for every element. So an alpha, as the formula implies, should be in between 0 and 1. So let me just declare alpha before the loop here to be, what did I use? I think I used 7.7. And now the cells are gonna coordinate. You can see that they're still struggling, but they are now more cohesive. Oh, I forgot something. They are not actually cohesive because, okay, I updated bias, but I didn't update the force that we are applying. And again, the finished product here is available already on the Google Drive. So I'm just gonna copy over the lines that define the force from the start, and I'm gonna paste them below the calculation of the new bias. So bias is going to be updated, and then we are using that bias to update the force. So this 20 here is just the force magnitude that we are applying. You could do more, that's fine, or less. And now should look something like this on the left. They are trying to go to the middle, but they are having difficulties. And that's partially because the force that we are applying, its magnitude is not that big. But another part is that our contact energy from moving and moving cells is a bit low, so they are too sticky to each other. So if I bump that to 25 now, we can see that they actually can let go and fill in the void.
Juliano, that code is pretty complex. Can you just copy and paste that block and put it in the chat?
I can, if I come here.
I'm not sure how well the spaces and stuff will
Yep.
carry over, but
The only problem with that is that you can't cut and paste into the NanoHub editor.
Yep.
So the thing to do would be in the break to put that code in a zip file and have it uploadable for people.
That code is here, so maybe the easiest thing is just to.
So why don't you show people how to upload it? That might be the easiest.
I should just.
Somebody says that you say that they figured out a way to use paste in NanoHub. Thank you very much for discovering that.
Let me see if I can do copy and paste to outside the NanoHub with that. Copy here. Now.
Yes, you can cut and paste within NanoHub. The problem is restrictions between NanoHub and the outside.
Yes, we would need the cells to reproduce, but this is getting to the collective motion. That's a complication for later. Well, I needed to copy this over back here because bias is just as an element of the dictionary. It does nothing. It's just information. So we have to apply the force, we have to define this lambda vac XYZ. And it's XCOM rev, not rev. Yeah, so this line here updates the dictionary. This line updates the force. Yes, alpha should be defined within step. And while you copy, what I'm just gonna say is go over the force velocity relationship. This I wouldn't have you code anything anyway, but here we have the Tokyo 2020 Cell Olympics with a bunch of cells swimming. And because of troubles of COVID and cinematography, we can't see the whole of the lanes. Yeah, if you change the template, it goes back to 10 every time because the energy is actually defined here. So you need to bump up the moving energy here to 25. But yeah, the cells obey Newton laws, as do all objects that are not super massive or super tiny. But their environment is exceedingly viscous. So what ends up happening is that if a force stops being applied to the cells, they pretty much instantly stop. So what happens is that force is proportional to the velocity. But that's not the whole story, as we can see here, because there is a saturation. And that happens because force will only be proportional to the velocity while the change in energy of the system is sufficiently small. So in here, for the temperature and other parameters, until 40-ish applied force was linear, but afterwards, this relationship of the change of energy got too high, so it stopped the linear relationship and it saturates. And this simulation is also on that same Google Drive. I mean, it's all under student materials, demos for students, demos for day 2, module 2.1. And we have 2 zips there. And that's it for me. Sorry, I went a bit over time. And yes, I could help people that are getting stuck on stuff now. We go to a breakout. Error codes is it's usually easier for the person having an error showing their code. than you looking at my code. Well, you'd probably fit your parameters to, I don't know, the cell velocity in real life, or their mean square displacement in real life.
That's an excellent question.
Yeah.
Cell tracking is a tricky problem.
Yep.
And it's gotten much, much better in the past few years as AI methods for doing cell segmentation have improved. And Gilberto Tomas, who will be lecturing later in the course, has done a lot of work on the statistical analysis of cell trajectories. And so I strongly encourage you to take a look at some of his papers on that. Maybe I can share those in the chat. And also ask him that question because he's thought a lot about it. And TJ is also working with a group which is doing quite extensive cell tracking, and he might have some good ideas about that too.
Yeah, in my case, the one is healed. I mean, there are voids, and that's part of the homework because the cells aren't reproducing, so they, well, their initial volume wasn't enough to fill in the gap. And you could see what happens with changing alpha. You could keep updating alpha in each time step so that at the beginning it's pretty random, or at the beginning the cells really try to align with the velocity, and at the end they try to keep going straight, or vice versa, and see what happens. You could also make alpha be part of a cell dictionary, and in that case you could have each cell care differently of which of the terms. And another good idea usually is always to add a bit of randomness to things. So here I have two options that I thought of to add randomness to the new bias, which is, the first one is multiply the whole calculation by a new parameter beta and use that parameter beta to add some noise to the result. Or have three separate parameters with alpha, beta, and gamma, multiplying them. But in that case, alpha, beta, and gamma summed up should be one. Otherwise, your bias will not be unitary anymore. So it won't only define a direction. It will also do other stuff.
Okay, we'll take a 10-minute break and we will start again at 12:15. Thank you, everybody. Okay, everybody, welcome back. I think we'll wait just another minute or two to make sure that the people who are in the breakout rooms have time to finish their questions and come back. I appreciate your patience, but some of the people who are in the breakout room are actually the people who are supposed to be presenting now. So we have to give them a minute to come back. Okay, thank you. I'm sorry, but I want to make sure that everyone who's scheduled to present has the opportunity. You're welcome to go back to the breakout room in a minute once we're done with the presentation. Okay, well, like to welcome everybody back. This is Module 2.2. What we have lined up for Module 2.2 is going to be some quick lightning talks by people presenting ideas for hackathon projects. And then we're going to follow that by a presentation of TJ, who is going to be talking to us about a COVID model that he's developed that is available online on NanoHub. And I think that would be pretty interesting for a lot of the people in the process. So I want to remind you again, this is Module 2.2. This is being live streamed and recorded, so please be aware of that. We're going to start out with Hackathon lightning talks. Then we are going to go to general discussion of Hackathon. If you have an idea that you want to bring up that was not presented before, that's fine. We have some ideas and suggestions also that we can help people with. And then we'll go on to TJ's presentation. So for the lightning talks, the ones that I have received are as follows. And so please be ready to speak for a minute or two about your talk. And people are welcome to ask questions and discuss a little bit. I want to make sure we get through everybody's ideas before we have to break. So to begin with, is Havid there? Would you like to tell us a little bit about your project concept?
Hello, everyone. I'm Javid. Yeah, my project would be about mechanobiology in the serial to mesenchymal transition, which is useful for calcific aortic valve disease if we identify and model this process. The thing is that this process happens when the endothelial cells start to delaminate from the monolayer and they lose their adhesion from cell to cell. Then they start to erode the base layer and degrade the ECM, and they go inside the ECM. So the transition happens there, and it can be problematic and cause the calcific aortic valve disease, causing some contributing for the formation of nodules and stiff valve leaflets. So in this project, I think that CompuCell can be a good software to develop the model. So I will be using the PD and OD solvers for diffusing fields, because some cytokines and chemicals need to be secreted by the cells and degrade the ECM, and or maybe for the chemotaxis purposes, the diffusing field would be useful. And also for signaling pathways, I think the subcellular simulations using tellurium and SPML language would be useful. So I'm a very I get involved in this project very early. I mean, it is recent for me, like several months or so. I don't know that much about, but if you have any questions, I'm open for that.
Okay, thank you very much. We'll make all of the emails available for people. I hope it's a great project. After all, Julio started out with a very simple model of EMT. That's a critical question in a whole variety of illnesses and also normal development. And so it'd be wonderful to see a team work on that. Great idea. Our next slide is AJ Chitnis and Abhishek Mukherjee. Do you want to tell us a little bit about your idea? And if you're looking for people to work with, please let us know.
Sure. Is it possible to run the movies that were in the thing, or it doesn't? Don't worry about it. There we go. So my lab works on this lateral line primordium, which is a group of cells that migrates from near the ear to the tip of the tail under the skin. And we've been modeling many different aspects of it during the past several years. So one important thing that we want to focus on is that as the cells move, they are reorganizing to form these little epithelial rosettes. And in the past, we had focused on the role of signaling interactions in defining spots of FGF activity which seed where these rosettes form in the migrating primordium. However, a number of experiments have shown to us that while signaling may normally play a role in where these aggregation of cells form, the mechanics of the system plays a big role in that, so that if the primordium slows down, you may have 3 or 4 rosettes present, but they will all come together and fuse to form a large rosette. I had made a simple model exploring this idea in NetLogo, where each cell is represented as just a single spot. But I want to take advantage of CompuCell to try and understand not just how they come together to form aggregates, but how their cell shape is influenced by these interactions. And I think a lot of what we've learned yesterday and today is like totally on point for what we are trying to do. My colleague Abhishek he has some experience in CompuCell already, and he has already been building some models. And in fact, he sent me some last night. But I think that what we are learning very systematically about the basics of what each of these different parameters do help us to much more rationally implement how we work our models. And eventually we want to simply, for the hackathon, help create a model which allows us to see how this tension between cells pulling on each other to try to make a rosette is counteracted by forces that are associated with migration, which try to pull cells apart, opposing this aggregation, and how the balance determines the size, number, and spacing of aggregates that form. So that's in a nutshell what we would like to do.
That's a very beautiful problem. I should say. My very first postdoc that I had more than 30 years ago, she was an expert in lateral line
oh, really?
genesis. And of course, people like Paul Kulesa have done work on neural crest cell migration that has some similarities to that.
Right.
So you have beautiful data and beautiful movies. Starting with the NetLogo version makes life a lot easier because you've already cellularized it and thought about those things. Julio will be teaching how to use links in CompuCell. And I'd strongly encourage you to get Julio involved because his work on somites without a clock
yes.
Was the kind of clustering aggregation due to junctional adhesion and filopodia, I think that you might want to be modeling here.
No, this is exactly what I was thinking during his presentation yesterday, because this is a evolving theme in biology where signaling provides some kind of reliability on the outcome, but it's working on the substrate of mechanics that can already achieve the job. And they work together to make a robust system. So this is exactly what we want to explore.
And there's also a simple simulation, which I hope we'll do in the chemotaxis element, which is based on simulations that Paul Kulesa did. If you have a diffusing chemical, it's rather hard to get the kind of directed movement that you see in the lateral line. But if you have a chemical that's a chemoattractant that's bound to the extracellular matrix, and the cells take it up and degrade it as they move, then that creates a persistent gradient that allows that long-range movement. Now, I'm not sure what's happening in this particular case, but that's also something that we could...
That is actually what's happening here. I didn't want to complicate this one-slide presentation, but actually this mass of cells is operating with two schemes. The leading cells are following a chemokine gradient that is self-generated because the trailing cells are degrading it. But the leading cells are also producing another factor, FGF, which is the cue for the trailing cells to chase after them. So they are working together to move.
So in some ways, this has similarities to angiogenesis, although it's a different cell type.
Yes, many similarities. Many similarities.
That's great. Well, I personally am very interested in that, so I hope I can help out with that. I'd love to see that simulation.
Thanks.
Any other comments or suggestions about that?
So I'll
Abhishek,
just-
does Abhishek want to say something? I don't know. If he's on. Go ahead, Julio.
No, I would just say that when I was doing my postdoc at EMBO,
Yeah.
Darren Gilmore, that you
All
may know,
right. All right.
he wanted to use CompuCell to model this,
Yes.
but his graduate student that was supposed to do that never follow up.
Oh, well, here we are.
But so I'm excited that this will be actually attempted and done, hopefully on CompuCell during the hackathon. So
Yeah,
I'm
and
very
I'm looking
excited
forward to
about that.
working with you guys.
Nice.
Okay, our next mini presentation is David. I apologize if I mispronounce people's name. Fagbemi. Are you there? I don't see David on at the moment, so if he returns, we can ask him to talk about his idea. The next one on our list is Farouche Farapur. Are you there?
Yes, I'm here.
Please tell us about your project.
Yeah, okay. I want to explain the two important phenomena that are important in this system that I am trying to model. So the first one is pancreatic ductal adenocarcinoma, abbreviated as PDAC, which is a highly aggressive malignancy. It is estimated to become the second leading cause of cancer-related deaths by 2030. Microscopically, PDAC, as you see in the left, consists of atypical tubular glands resembling medium-sized or smaller pancreatic ducts. PDAC microenvironment is very complex, composed of a minority of malignant cells within a microenvironment of extracellular matrix, fibroblasts, endothelial cells, and immune cells. Cancer-associated fibroblasts, abbreviated as CAFs, are one of the most significant components in the tumor microenvironment. Despite the challenges that CAFs make for the treatment of the disease, their existence in tumor microenvironment is now considered as an opportunity for a targeted therapy. Okay, this was a very short and incomplete picture of PDAC that I could present here, but let me go further and talk about the other aspect of the idea that you see in the right picture. Immunogenic cell death, abbreviated as ICD, constitutes a type of cellular demise that elicits an adaptive immune response. ICD has been characterized in malignant cells following cytotoxic interventions such as chemotherapy and radiotherapy. As you see, a set of damage-associated molecular patterns or DAMPs like ATP and so on are released by ICD that activate dendritic cells, which in turn recruit T cells into the microenvironment and thus start an immune response that can attack the tumor cells that are treatment resistant. Special effects play an important role in PDAC tumor microenvironment. So the idea is to model ICD-induced immune responses in PDAC with a hybrid model that simulates individual cells with an agent-based model, for example, cellular POTS model, and the immune mediators with ordinary and partial differential equations. That's it.
TJ, if you're there, I thought you might have some comments on this.
Yeah, this looks interesting. So maybe in the bit of time that I get with you towards the end of this module, we'll very briefly look at some related work that we've done, associated with modeling the immune system, both, in a local scale, doing spatiotemporal cell-based modeling, and then also with that being coupled to more of a systems level immune response. So, but Yeah, maybe we should talk about this, I don't know, on a break, or maybe try to catch up at the beginning of the hackathon, and maybe discuss this in a bit more detail.
Yeah, it would be great. Thank you.
Great.
For sure, I would learn a lot. In fact, I do not have a solid background in biology. I am a physicist by training. So if people here know more about PDAC, about, I don't know, the biology of this system, it would really appreciate it if they can also help us. Yeah.
Well, to speak to that, my background is mechanical engineering, so you're in good company.
And I'm a physics PhD too, although I've been doing biology for 30 years. I was the chair of the Division of Biological Physics of the American Physical Society for a couple of years. So I'm also a physicist in background. So especially because we have a very diverse audience, if there are groups of people who are looking for projects and who have special expertise, for example, if there's a cancer biologist in the room who'd like to team up, that's a great opportunity. So building teams, you could certainly do a hackathon on your own, but it's a lot more fun if you have a team. And so if you're not part of a team already and you're looking for a project, please consider these projects and let us know. Let us know, but let the people who are proposing them know whether you're interested or not. We'll have another discussion, I believe, on Thursday, where we could pursue this. So please do work on your own, trying to connect with instructors and with other class participants to build some teams. Okay, the next slide is from Hamed Fatoyimbo. Are you there? No, well, I hope Hamed will join us again and have an opportunity to present. The next slide was Megan.
Hi.
Is it Haas or Hassa?
It's Hossie, but yeah.
Hassi. Okay.
Yeah, so my idea is an agent-based model of muscle regeneration following an acute injury. So my lab focuses on looking at muscle, and we've done previous models similar to this in Repast, but with my experience in Repast, I'm interested in switching to CompuCell because of some of the advantages in modeling cellular systems. So specifically, in terms of this model, we would have some acute injury that would cause damage to the muscle fibers, and then you would have this inflammatory response, so like neutrophils and macrophages moving in. Then over time, you would start to see like satellite stem cells move in and proliferate, and then depending on the degree of injury, you could start to see fibrosis if there were too many fibroblasts or prolonged inflammation. And then over time, this would heal, the satellite stem cells would help regenerate the muscle fibers. So I'm also interested in looking at some other components of muscle regeneration, and depending on what my team is also interested in, maybe including like different sex hormones to see how like estrogen or testosterone would impact muscle regeneration. In terms of computational approaches, a lot of logic-based or literature-derived rules. I would need some type of equations for diffusion of the cytokines or the hormones, as well as ECM remodeling and having the cells sense their neighbors to determine movement based on like cytokine levels. And then expertise for partners, really just an interest in the model and the system, any experience with agent-based modeling or cell diffusion dynamics or experience in Python or CompuCell would be great. And my email is on that slide.
That sounds great. When you have an existing model, that makes it much easier, of course, to re-implement. And again, I think that's a great project. It would be wonderful to see that come to fruition. So please And I think what TJ will show us in terms of the immune modeling might be helpful as well. Great, thank you very much. The next slide is it Yajun He?
Yeah, hi. Yeah, my last name is pronounced like her.
Okay, sorry. Thank you.
Okay, that's okay. And I think the tentative title of my project is called the Computational Modeling of Gene Regulatory Network and Trajectory of the Developing Midbrain Dopaminergic Neurons. And the plot on the left side shows the dopaminergic neural development in mouse midbrain from prenatal to postnatal, and the development the progress involves like the dopaminergic lineage specification and its differentiation and maturation. And this progress has some spatial characteristics, and like the progenitor cells will give rise around embryonic day 8, and they will proliferating and differentiating into the neuroblast. And around day 10.5, the neuroblast will migrate radially from the ventricular zone to the marginal zone, and where they will mature into the dopaminergic neurons. And after the maturation, they will migrate into their final destination and project to 3 distinct areas. And I think each of the major development process is orchestrated by a number of morphogens secreted by the surrounding tissue, and like the Sony Hedgehog and FGF8 and Wing1. And in response to this morphogens, a set of transcription factor plays a vital role in determining the cellular fate. And we have derived a gene regulatory network using some bioinformatics tools from the single-cell RNA sequencing data, and we trimmed it by cross-validated with the existing literature. And so my goal is to construct a dynamic model of this gene regulatory network. I'm thinking starting with a basic Boolean model, and in the future, I want to construct an ODE-based model. And the ideal will be like cooperate with spatial modeling to simulate how the neurons will migrate during the whole development in the midbrain. And yeah, that's it.
Okay, that's great. You'll hear about Boolean network modeling on the last day. TJ will be presenting some of that. Actually, I guess on Thursday he'll be presenting that.
Okay.
And these are good or wonderful problems in a sense. It's got some similarities with the neural crest. I was just saying neural crest, the lateral line cell migration in terms of some of the issues. So I think that's great, and I hope that we'll be able to help you come up with a simulation that we can be useful.
Yeah.
Okay. Okay, our next talk is Aaron Hodgkinson. Are you there?
Hi, Jim. Can you hear me?
Yes, that sounds good. Thank you.
Ah, great. Yeah, I assume, given that the text is posted over the video, that the video wasn't working. But there was a video in the lower right-hand side here. It's not necessarily a problem. The basic problem is though, yeah.
I'm sorry that when it converted to PowerPoint, we weren't able to figure out a way. Do you want me to have you screen share? I can have you screen share for a second if you'd like to show the video.
Sure, if that's easiest. My Zoom has disappeared. Okay. Can you see my keynote? Yeah, so the basic problem is that you have this cluster of cells at the beginning of zebrafish embryo development. And during the process of convergent extension, where you have the sort of blob of cells turning into a fish very early, you have some cells at the front of the invading cell front, which are communicating to cells very locally a Wnt activation signal. And over the course of this epibole, they call it, the conversion from a blob to a fish, essentially, you have a distribution of Wnt signaling across the fish, despite the fact that the signaling was very local. So what we're interested in is the interplay between this convergent extension process and the distribution of the Wnt signal across the tissue. But also, if I can show you this video, what you can see is this is a Wnt signal in a zebrafish embryo. See if I can make it a bit bigger. And you can see as the epibole is taking place, you get this sort of noisy signal. So you have a very nice Wnt gradient, but you also have these noisy cells. And the biologists I work with say that apoptosis should play a very small role in correcting this gradient. But a lot of papers say that apoptosis should play a major role. And so what we want to try and find out during the hackathon is what kind of a role apoptosis is likely to play, major or minor.
Okay. So that's a fascinating problem. We've worked with a number of teams that are interested in various aspects of zebrafish epibole. And so we'd be very interested in trying to help you with that. I don't know if there anybody else on the team here who has expertise that's relevant. There are a lot of complex issues in epibole because so much is happening at once. And so I think one of the key things with a project like this would be to try to define at least an initial set of questions that are achievable with a relatively simple model. But
so
I know TJ is thinking about these issues a fair amount these days. So
okay, great.
No, go ahead,
One of
please.
the things we had considered is potentially modeling on the plane just to begin with before going on to the sphere.
So definitely, but again, it's great to see. Last year's CompuCell course was built around COVID and viral infection modeling, and so it's great to see so many developmental biologists back. It's really nice. Okay, thank you very much.
Thank you.
Our next talk is János, is it?
Yes, I'm here. Thank you. Do you hear me?
Yes.
Perfect. Thank you. So I'm interested about yeast colonies, the growth of yeast colonies. Here in the image, we can see how a colony of yeast cells grow and develop. And technically, it looks like it's a 2D slice of the 3-dimensional colonies. And actually, we see it starts growing like a ball, a balloon. But after a time, the middle part, the top part, is not growing anywhere, only the sides. So I'm interested in constructing some model about these processes. Then technically the middle part of where the colony starts growing like a ball or a balloon, but after a time the central cells don't have enough oxygen, nutrients, so they don't divide anymore, while the cells at the edge of the colony still dividing. And the base of the colony and the top layer somehow separated, and some cavities appear around the edges. And as the colony grows, cells can divide inside these cavities, and the colony grows also at the sides. So at the end, some Winkel structure, some complex structure could arrive. That's what we see in microscopic images. And I'm interested about how to use CompuCell to somehow mimic these kind of processes. I know it contains some gradients and also biophysics between cells, attachment, detachment, things like that. So that was my idea technically.
Okay, I think Julio will be showing some buckling simulations later in the course that could be used, and some of the models of nutrient-limited growth might be relevant. So combining a nutrient-limited growth model with a mechanical model of buckling might very well be reasonable for a project of that kind. Whether you could simulate quite as many cells as you have in some of your pictures, I don't know with that level of detail, but I think you could probably at least do a 2D simulation of what it looks like. And that would be reasonable. Again, people are welcome to break in and make comments if they have suggestions or ideas.
Okay, thank you very much.
Okay, our next slide is from Iman Kafian Atari.
Yes, here I am. Okay. Hello everyone. My name is Iman, and my project is about modeling the impact of cartilage degeneration on its physical properties, such as its optical response. So what I would like to do in this summer school is to develop multiple agent-based model of cartilage and try to simulate degeneration through these models. And later on, I will incorporate these degenerated models into radio transport equation and estimate light propagation in the tissue, and also versus the degenerated tissues. And we use these changes or alteration between light propagation in healthy and degenerated tissue as a marker for assessing the pathology or degeneration in the tissue. Yeah, and that's the whole idea of this study. And this hackathon project is actually about developing the agent-based models of cartilage. I would like to start with the basic model, which only comprised of solid matrix and water of the tissue, and then later on eventually progress to more advanced models that capture the composition and the structure of the tissue fully. Yeah, so that's it.
I think that that's very promising. I remember a team started a cartilage model many years ago with CompuCell. They got quite far, but somehow it was never finished and published. And I would love to see that cartilage, those cartilage ideas work. The idea of the breakdown of the extracellular matrix is critical, and I think it's very appealing. I hope again that you'll find some people to work with and some support for that. That's a wonderful project.
Yeah, hopefully we'll show interest in this project too.
Okay. The next one is Maria Raffo. Is Maria here? Maria Del Valle or Raffo. She wants to tell us.
Chat.
Oh, well, I don't mean to put you on the spot. Infectious disease modeling is certainly something that we can do. And if you'd like to talk to what is your native language? We certainly have Spanish and Portuguese, so Brazilian Portuguese speakers in the room. So if you'd like to talk to somebody, that's certainly possible. Spanish, I believe we have at least one or two Spanish native speakers. So please connect with us and let us know what we can do to help out. Okay, I don't mean to put you on the spot in terms of presentation. Okay, the next one is Lorenzo. You want to speak up a little bit?
Hi. Okay, can you hear me? Okay, my project is about implementing a multi-scale simulation of endothelial cell. And the idea is to study the strength of interendothelial cell junction, spatial constraints, so how cells are disposed in a monolayer, and notch activity and proliferation. So this idea came from some work we have done to extract data from imaging of endothelial cell monolayer. And we built this endothelial cell profiling tool so that we can extract data from a lot of images, and that allows us to account for the experimental variability and also some biological variability. And analyzing this data, we find out there was something that did some heterogeneity in notch signaling, which didn't seem to fit with the current understanding, or perhaps we want to understand a little bit more on how the rules and the regulatory network with notch activity can give us as a result this heterogeneity. And you see it in the image in the middle. This is a kind of map of an ICD, and is obtained by this endothelial cell profiling tool, and these are experimental data. So our aim was to implement a CompuCell simulation, which could account for the current understanding of notch signaling that has been already implemented to some extent into antimony or SBML or these kind of tools. We have done a little bit of work on that, and we have created a simple initial CompuCell simulation which can account for this and allow us to import our maps of cell and data into CompuCell and try to use the SBML model to extract data from this. And what we want to do is to generate synthetic data and see if we can actually reproduce those data that we have in vitro with different endothelial cell line, and figure out a little bit better about the biology. So in general, for the hackathon, the idea was either or both, to refine a little bit our simplistic CompuCell simulation, so perhaps introducing proliferation or some evolved version of the cell junctions, and to learn and perhaps implement a little bit of parameter scans, sensitivity analysis, and look if our SBML model can really reproduce the data, and then we will analyze back into our ACP tool to analyze this data comparatively and see what we can learn of the biology of the system. And one main question that is very interesting to us is which is not signaling in the same endothelial cell monolayer effectively able to stabilize. So is that possible that all the cells become positive or all the cells become negative, or there will be always some kind of movement there? And what is the biological meaning of that if that occurs? Thank you.
Okay, and the last one is this unclaimed slide. Does this slide belong to anyone? Somebody posted this very nice slide with no name on it. Using PhysiBus, modeling of cancer, breast cancer invasion. Does anybody recognize the slide? And if so, would they like to speak about it? No? Okay, I will. It's an interesting slide, so it'd be nice if we had somebody to claim it. But okay. Okay, I think those were the main hackathon ideas. There are plenty. There would be plenty of work there for everybody in the group to work on. But if some of that didn't appeal to you or didn't strike your fancy, there certainly are many other things that one could work on in the general area of wound healing, cancer tumor growth, the kind of viral model that TJ and immune model that TJ will show in a minute. We heard a nice presentation on diffusion limited growth and buckling in yeast. So that would be very similar to a biofilm model. We've heard talks about convergent extension already, so that one is pretty well taken. Joshua has done some work on earlier stages of embryogenesis and blastula formation that could be used to extend. And there could be some interesting simulations of collective movement with cell crawling based on the simulations that Gilberto will show. There also are a bunch of software tools that would be nice to have in CompuCell. People asked about sensitivity analysis and parameter identification. We have some tools, but one could make those more specific. We heard about people who have some AI expertise, so it's possible that somebody could extend those ideas. And somebody asked about image export and import. There definitely would be room for tools of that kind. CompuCell is designed to be modular and extensible, so it's relatively easy to build plugins and importers that will work with it. If really none of that appeals to you, and there's been such a variety already, something that we could do is, I recommend, especially if you're starting out, is to think about replicating an existing model. We heard already a talk about the lateral line and having a model that was written in that logo. If you have an existing model that's written using an agent-based formalism, it's much easier then to translate that into a CompuCell implementation. One could take an existing CompuCell model and extend it, or potentially take a model that was written for another environment like Morpheus or Physicell or NetLogo and convert it to CompuCell and see, learn about how that works. And so if anybody's interested in those kinds of problems, please let us know. I'd like to give us just a few minutes to have conversation. If there are any questions, comments that people want to discuss, questions for our speakers. If not, well, let me give you a few minutes for that. And then we'll move on for TJ's short presentation. Does anybody have any suggestions? Somebody says, do those who speak want us to contact them by email or Slack? My suggestion is that you try Slack and email. I will post the email contact lists for people. You can also message people while you're in the Zoom. So you can send messages using the chat feature. So that's a possibility. So let us know what we can do to help out in terms of connecting people. Again, we'll have a meeting on Thursday where we'll do a follow-up on this and try to help make sure that people are developing their ideas in teams as we go forward. Does anybody else have anything they want to bring up at the moment? I think it's a great set of projects. If you want to make a Slack channel for each project, that's fine. Definitely you're free to do that if that's helpful. It says, is it possible to join as an observer and not an expert? Teams always need people with all sorts of different expertise. And being a beginner is an aspect of expertise too. So don't be shy about approaching a team that you think is interesting and ask if you can participate. There are always things to do, whatever your experience level. Okay. Any other comments or questions? No. Please feel free to ask during the breaks or before or after. One of the reasons we have that half an hour before we start and half an hour after we start is to have more discussions of this kind. So please, and if people want to go into a breakout room to discuss a team project, please let us know. I can certainly create a breakout room for people to have a conversation through Zoom. So that's definitely an option for people. Okay, well, I thank everybody for doing their presentations. TJ, are you queued up to go ahead?
Yes, James, I'm ready to go. All right, well, hello, everyone. For those of you who joined us on Sunday for the Python crash course, it's great to be speaking with you again. For those of you who I haven't had the pleasure of meeting yet, my name is TJ Seago. I'm very happy to be talking with you today and working with you throughout this week. On Sunday, we all got together and did 8 solid hours of Python crash coursing, which has since been described to me as drinking from a fire hose. But we all powered through it. It was an excellent experience. And so today I get to talk a little bit more on the biological side and the modeling side, which is nice. While I do a lot of work on the development of CompuCell per se, I also do a lot of modeling, as James has mentioned, much to do with viral infection modeling and immune response modeling, and to do with some of those aspects of host pathogen interaction and so on and so forth. So I'm very glad to be speaking with you about that and get to share some of those projects and ideas. To that effect, most of what we're going to do for the rest of our time in this module is going to, in one form or another, revolve around this paper that is shown on my screen right now. And I'm going to put a link. I'm actually going to put a couple of links in the chat. I'm going to start with the link to this paper. So as James mentioned, when we were doing the workshop last year, you know, we and many other people in the sciences pivoted in one form or another to try to somehow contribute to the pandemic. And this paper is one of the results of that work. In fact, at that time, we were sort of in the weeds on working on this particular project, which is about a framework that's built within CompuCell to be able to do very complicated, elaborate modeling on viral infection and immune response. And so we're going to sort of revolve around some of the work that was done to complement a lot of either what we've discussed so far in the workshop or what we will discuss in later modules throughout the week. So that first link is a link to find the actual paper. If we're not going to get into a lot of the details, and it is a quite lengthy paper with a lot of detail, we're going to omit most of it. And so if you do find yourself interested in wanting to know more, that we will skip over for now, you can use that link to actually review the paper. I'm also going to include a link in the chat that's actually on the slide, which is the GitHub repository for those of you who may be interested. This is the repository to the actual code base for this platform. And in one sense, this is made available for transparency and for reproducibility. On the other hand, this is also specifically designed for people to actually grab this code and potentially contribute to it, to use it for their own purposes, for all sorts of things. So I want to make sure that that's available to you. You can follow that there for those of you who would like to pull that code and maybe do a little work on it and make some contributions yourself. We not only encourage that, we invite it. And then lastly, and this link I will post in the chat again because we will come back to this, but for now, for those of you who like to do things while you listen, you can go to this third link. This third link is a separate NanoHub tool, much like the one that we've been working in for all of our CompuCell work. But this is the actual NanoHub tool for this viral infection framework as it was published in the paper. So you can follow that third link. We are going to come back to this. If we have time, we'll make time, at least a little bit. And we'll actually open this thing up and watch it go. In the meantime, some things that I would like to do in the next half hour or so is to actually walk through some of the processes of abstracting biological objects or processes that we may be interested in, and sort of walk through, at a very high level, some of the logic of actually, generating things like mathematical models, agent-based models, to represent the things that we may be interested in actually modeling and simulating. I want to at least provide some sort of sense of what, a fairly elaborate research grade CompuCell simulation actually looks like. Again, not diving into the details, but to give you a sort of glimpse of kind of a terminal point of some of the, basic things that we've learned so far and will continue to learn this week. And I'd also like to send you off with a sense of what CompuCell enables, in terms of more of the social aspects of modeling sciences. In computational biology, we can ask, can we actually use our simulations in more of a broader sense? Can we, say, make them available to others for them to be able to use in CompuCell? Or can we work with others to work on the same simulation, but in different aspects? Does CompuCell support that? And we'll show that this framework demonstrates that it does. And in my opinion, quite well. And so we'll take a look at that. I'd like to start by sort of motivating, the activity of generating these models for COVID. When we came to this problem in the first place, in terms of COVID, we asked these questions for infectious diseases, these types of questions for infectious diseases and developmental diseases, asking, very basic questions about this particular problem, like, variability in patients. Why do some people get sick who are exposed, but other people who are exposed don't get sick? What are the responsible mechanisms for that sort of variability? What causes complications during the progression of an infectious disease? And then more applied questions like, can we generate predictive models and, modeling and simulation technologies that would enable us to, say, be more effective in delivering therapeutics? Can we do, as James has mentioned a few times already, can we do, individual personalized medicine? And if so, can we develop things that can further that cause? In this case for COVID, but, of course, for, other diseases. And in the case of CompuCell, many times we find ourselves asking things like, can we describe, these more emergent events like getting sick in terms of, more molecular level perturbations, cellular level mechanisms, things that could actually potentially be targeted, with drug therapies and so on and so forth. So we're going to, sort of view all of this with that sort of motivation. And then want to do this activity of, sort of breaking this down into some very basic concepts. So when you have something like, a SARS-CoV-2, COVID-19, what occurs in this event of exposure and infection is that a virus is introduced to the respiratory tract. It infects an epithelial target tissue, and then things happen after that. And we ask when there is, say, an immune response to this infection that is actually going to progress. the response is an immune response. And so then we ask what are, sort of the driving aspects and, what are the characteristics of those aspects. And in this case, when we have something like a local infection in a respiratory tract, this consists of a small neighborhood of cells. There is then a corresponding whole body response that is the immune response. So something local that occurs that then results in a response throughout the body that we'll just generally refer to as being a systemic response, in this case, a systemic immune response. And then lots of other peripheral mechanisms that we may or may not be interested, depending on what we're looking to model. In this case, things like transport throughout the body, whether, blood, lymph, or air, or say if we're interested in, complications, say spread to specific organs, to specific tissues, to say investigate more critical cases of an infection. And for people like me, say maybe interested more in specific aspects of the immune response. So we have a mechanism that's involved in our total event of a COVID-19 case. And, through that, we can actually dive into the details of specific mechanisms associated with it. In the case of the immune response, whether it be the innate or adaptive immune response, and some particularities, associated with all of those processes. And so in the case of this project, we adopted the strategy of starting very simple because cellular modeling per se can be quite complicated. And then applying that to a very complicated problem like an infectious disease and an immune response, very complicated. And so our strategy to start was to begin with a local patch of epithelial tissue that would be exposed to a virus, and then start to build in some of the mechanisms of interest into that model around that paradigm of having a local epithelial patch. So in this case, we assumed that this is a primary infection. So this is the first time that our host has been exposed. And you can see what one of these simulations sort of looks like on the right. We'll go into some of the details a little bit. But in general, we're talking about an epithelial patch that gets infected, and then lots of events follow. And like to sort of show you the connection of going from, observations and current knowledge about the biology and making the translation to the actual model objects, whether they be CompuCell specific or just general modeling assumptions. And so in this case, if we're considering, say, a lung epithelia, lung epithelial tissue, so we have some sort of quasi two-dimensional patch. If we're looking close enough at our epithelial tissue, there can be lots of different cell types. When we generate a model that corresponds to that for a very local patch, we have something like a two-dimensional epithelial tissue. So this is actually a screenshot of one of the simulations. Just looking at the epithelial tissue, you see this is a two-dimensional simulation. And in this simplest case, just a uniform layer of epithelial cells. There's a lot to do with the actual replication of virus. When a cell is infected, there's a lot that goes on inside of that cell to produce more virus. A virus goes into the cell, things happen to produce more virus, and those resulting virions then leave the cell to go on and propagate the infection. Now, in a what may seem like a quite complicated model, in a very simple case, we can very readily in CompuCell, say, generate an ODE model describing the various phases of the viral life cycle inside of an infected cell. That model being shown here on the right with the little schematic of some of the associated mechanisms of the viral life cycle. And this is simulated in each individual infected cell. So moving through the various stages, and that's after virus gets inside of a cell, leading to release of more virus outside of the cell to do more infecting. Now, the immune response is wonderfully complicated in so many unbelievable ways, and this is by no means an exhaustive list, but there's a lot that goes on, from initial exposure and the innate immune response, phagocytosis of virions, the interferon response, all the way to antigen presentation, B cell maturation, T cell recruitment, antibody production, so much that goes on. And so in the case of this initial project, with just a total acknowledgement of a blatant simplification of mostly misunderstood, a mostly not understood system, we opted to have an immune response in our model that does relate aspects of a systemic response to our local patch, but to do it in a very simple way. So we have things like signaling that occurs from our local patch that then does some sort of recruitment that brings an immune cell type into our local patch. So signaling out of the patch, recruitment of an immune cell type into the patch. And those immune cells can do different things that represent, relevant mechanisms that, say, natural killer or T cells will do at a site of infection, like contact-mediated killing, say, neutrophil release of reactive oxygen, things like that. And so in this case, for our preliminary work on modeling the immune response in this framework, we elected to use an ODE model that represents the total inflammatory state of the infected organism, using some convention where we have this very abstract variable S, and we say that when our variable S is greater than 0, the species is in an inflammatory state, and when it's negative, it's in an anti-inflammatory state. And we couple the dynamics of that variable to signaling in the local patch, where signaling in the local patch can drive the system into an inflammatory state. And when the system is in an inflammatory state, that then leads to recruitment of immune cells into our patch. And likewise, pulling immune cells out of our patch is due to the species being in an anti-inflammatory state. So a lot of simplification, but reducing this to something that's much more manageable in an otherwise still very complicated system, at least to a workable degree, where we can start to generate simulations that actually have recruitment and a local immune response. And then there's lots to do with damage and scarring and tissue recovery. Epithelial cells die through lots of different means during COVID. And there is a lot to do with recovery, most of which we did not treat in this particular work. The farthest that we really took this was that cells can die. And we did a little bit of work on showing different ways that we could model recovery. But in general, when you see an epithelial patch and you see these black cells in here, so this is damage, dead epithelial cells in an epithelial tissue, whether it be due to viral replication or some action of an immune cell. Now, something that is both very interesting and also very complicated about this model and models like this is that you consider that the survey that we just did goes all the way from subcellular viral replication all the way to a systemic immune response, and all built around a CompuCell simulation, considering individual epithelial cells. You have a pretty broad span on a multi-scale model, going from subcellular kinetics to a whole body response. And probably importantly to point out here that, when we talk about these viral replication kinetics and we have a viral replication model for an infected cell, we have to consider that viral replication model is being simulated inside each individual cell. You go look at any cell in simulation, and you'll learn how to do this starting tomorrow. That you look inside each individual cell, and you'll have one of these running models if the cell has been infected. Whereas there is a running ODE model of the inflammatory response. So it's a detail there that I think we'll be more familiar with by the end of the day tomorrow. And this is what it looks like, and this is a schematic of all of the model modules that consist of the entire framework. As I mentioned, this is a pretty long paper with a lot of detail, and we're not going to dive into much of the details on any of this, hence the paper. And of course, Josh, Juliano, James, me, any of us would be happy to talk with you more, about some of those details, outside of the modules. Now, we can do a lot of interesting things to explore the sorts of questions that were mentioned towards the beginning of our time today, about asking questions, say, about molecular perturbations and how they may affect sort of emergent events, emergent properties. In this case, and in this paper, we go into this quite a bit, about simulating a drug therapy, and in particular, studying the effects of both the potency and the timing of a drug therapy, with respect to initial exposure, asking things like, what is the effect of administering a drug earlier versus later, after exposure? And how are those effects, say, changed by the potency of a therapy? And you can see what this sort of looks like here on the right. So this is actually a parameter scan, varying the time in simulation when an antiviral therapy like remdesivir is administered. And then also while varying how strong that therapy is, how potent it is. Here, moving from left to right, we have earlier treatments, the dashed line showing when in simulation the therapy is administered, to very late drug therapies, and then from lower potencies to high potencies. The potencies here being reductions in the rate at which new virions are produced by infected cells. And as you might expect, and James surveyed this on day one, earlier treatments are more effective, stronger treatments are more effective. And so I won't dwell on that, but this is what this looks like in the context of these individual model modules. Now, one of the other cool things about this framework and what can be built within the CompuCell environment is the ability to actually develop these simulations in a way that can be improved by others, that can be used to facilitate sharing of models or collaborative development. And we talked about this, I think, in the chat yesterday a little bit about what is the point of having multiple steppables. I can't remember exactly who asked that, but I was very glad to see the question asked, because why not write all of your models of a simulation in a single steppable? And one of the answers is that if every model is implemented as its own steppable, then it becomes possible to use the steppable that implements that model in lots of different simulations. And you can see an example of what this looks like in this schematic. So this framework is divided where each steppable implements a particular model, and then they're all assembled into the overarching simulation. But nothing prevents someone from coming along and, say, not using all of the steppables that we implemented. And nothing stops someone from coming along and writing a steppable of their own that implements some model of their own, and then also implementing that in their own simulation that otherwise uses everything that we had already written. And this framework is specifically written and designed in such a way to actually facilitate that. So it is possible to write things like modeling and simulation environments within CompuCell where lots of people can contribute simultaneously, in some sort of collaborative or, large scale, cooperative project working at different aspects of a broader problem. If, say, you are interested in developing better models of viral replication, but I am interested in building better models of the immune response, well, each of those are implemented in different stepables, which means that we can work on the same simulation by improving our own respective stepables and all still contributing to the same overarching computational model. Now, I don't want to spend much time at all going through the details of code, but this is what this looks like. This is how this published work was done. This is stepables just like what we've been writing and what we'll continue to write throughout the week. But in this case, this is actually leveraging some of the additional functionality that comes from using Python itself in CompuCell, in that this is actually taking stepables that have, say, been written by some imaginary group and then adapted by some other group. In this case, to improve the recovery model. So for those of you who were with us on Sunday, you remember me talking about inheritance, and we spent a lot of time on working with classes. And this leverages some of the basic aspects of class hierarchy and inheritance in Python, where one group can come along and inherit the models from someone else's stepables and then improve them. So model sharing, not only in the sense of making your stepables available to people, but making them available in the sense that they can directly use them and they can directly improve them or add on to them or use them in ways that maybe you hadn't thought of. Now, using this capability, we've already supported a number of projects and are continuing to support projects that have, in one form or another been constructed from this framework. James mentioned yesterday about one of our projects being modeling the immune response to influenza infection. This project, which is shown here, maybe this looks a little familiar. So we've got another epithelial sheet. We've got some infection going on, but in this case, not say a highly simplified immune response with one immune cell and some major abstractions, but instead now we're talking about explicitly type 1 interferon. In this immune cell layer, we're talking specifically about macrophages, NK cells, CD8 plus T cells, and all of this is constructed around this framework. And this same basic principle of having a local epithelial patch in this schematic, everything inside this dashed rectangle those are all objects that are explicitly represented in the local epithelial patch. And then everything else is a part of the systemic response, all of that being constructed in this framework, just substituting out the immune system model stepables, changing the way that the viral replication model stepable works, and implementing them in such a way that we can generate a multi-scale spatial model of a previously calibrated network model of a lot of detail of the immune response. So I want to fast forward through some slides because we only have 10 minutes left. And what I would like to do is migrate over to the tool. So I'm going to put in the chat again the link to the NanoHub tool. And what I would like to do is, even for those of you who have been working locally with a local installation of CompuCell, we have the code available through the GitHub repository. Here's the link to the NanoHub tool. The source code for the framework can be cloned from the GitHub repository if you'd like to work with this on your local installation later. But for the sake of time, Farnish asks, is this influenza paper published? It is under review currently. And we do expect that hopefully to be published very soon. I'm quite excited about that and excited to build from it. So for the sake of time, let's go with the NanoHub tool for now. Be happy to work with people who would like to get the source code from the GitHub repository and use it in their local installation. Later, we can maybe talk in private chat, or you can email me, and I can provide some basic instructions on how to do that. So following the link in the chat, it should take you to a familiar looking page. Should look like the CompuCell v4 main tool. This is a different tool, same mechanism to launch it. There's a launch tool button. I'll give everyone a couple seconds to get here. And we're going to click launch tool. And we're going to get a tool that looks like the one we've been working in. It will be an instance of a CompuCell installation. It will have player, it will have Twedit, but it will be preloaded with the simulation that was used in that paper. So if everyone's here, let's click the launch tool. I may get faced with a sign in thing. Nope. So I'm in. Let's cross our fingers for more great performance from NanoHub today. And if you're with me, then you should see CompuCell starting up. And one major difference from maybe previous experience so far is that in this case, there are a lot of windows. So to start, let's just go ahead and go to the main menu bar. Let's go to window and let's tile. So window and tile. And if you'd like to just focus on some of the spatial results, which I would strongly recommend, you can just minimize all of the windows that have scalar transient data being plotted, because in just the few minutes that we have, I'd really rather focus on just looking at the basics of the actual spatial distributions. So for everything except maybe the plot that shows a cell field, let's just minimize it. And let's get 2 graphics windows going. Should have 1 cell field, probably showing the immune response. It should say cell field and then have an XY coordinate of 1. This is a coordinate looking up and down at the sheet. In this case, in our simulations, this is a quasi 2-dimensional simulation where at z equals 0, we find our epithelial cells, which you'll see here in my screen when I change this coordinate value to 0. Now I'm looking at lots of blue cells. These are epithelial cells. And then at z equals to 1, on top of our epithelial sheet, is where our local immune cells do their business. When we bring an immune cell into our local patch, we say that in our scenario, we're looking at something like the top of the epithelial layer on which an immune cell is placed and can move around and do things like contact-mediated killing. So if you have one graphics screen showing 1 cell field, then let's create a second window. You can do that with the very last of the icons in the second row in player. Same row as your play, step, pause, and stop buttons. It'll be the very last one. Create a new graphics window. And to get something like what I'm going to do, you can have both of them showing the cell field. One at z equals to 0, just setting the value in the numerical field, and the other at z equals to 1. And if you're with me, just hit play you can watch this go. We have 4 minutes left, so we're not going to be able to spend a lot of time on this. Although I would encourage you to say play with this more. The slides that are available, I included a lot of activities for perturbing this system, which you can do in the steering panel that's included in this tool. So one of the things that maybe you saw me minimize or you noticed when your tool launched was that there's a steering panel that's pre-configured with 3 different items. Each of these is coupled to a critical component in the model that can have very noticeable effects on the outcome of the progression of infection. So we saw in the movies and the slides that infection will start with a single infected cell and will propagate outwards. There are a lot of parameters that affect different mechanisms that will either discourage that spread of infection, if not stop it altogether, or strongly encourage it and cause it to propagate more or faster. Each of these is set up to be able to let you do computational experiments by doing variations of parameters associated with internalization of virus, which is this first entry, the rate at which immune cells are recruited to the local patch, that being beta delay multiplier, and then the rate at which viral replication occurs inside infected cells, the one that we varied in our drug therapy simulations that we glanced at very briefly, that being our max multiplier. The slides at the end of this module have a whole set of activities of things that you can vary and see what the corresponding effects are on the spread of infection. And those slides also include locations to actually find these parameters in the source code. Now, some of the activities are actually not supported by this steering panel, in particular, varying mechanisms associated with cell death due to the release and exposure to reactive oxygen. In that case, there's not a steering panel item configured for that, specifically to send you to the source code. So you can open TWEDIT in the same way as you would in the normal tool. And if you look at the slide, you'll find screenshots of the source code to actually find the corresponding parameter in the model parameters file. So you can go through and programmatically do variations as well. The hope is that that motivates you to actually dive into the code and maybe start making some manipulations of your own. Because again, should you feel so inclined to actually do some model development with this framework, the GitHub repository is specifically set up to facilitate that, in the sense that if you were to, say, develop something that you would like to share with the broader community, dare I say publish it, we would be happy to host it on our repository as an importable model module that would be distributed along with the broader framework. So there are instructions on the repository as well about how to do that. Again, refer to the slides for the actual activities that I've set up. Say we're out of time. So James, I'll turn it back to you. Unless there are any questions that maybe we can work through before we break for lunch.
No, that's fine. There was a question about pharmacokinetic modeling or diabetes modeling with CompuCell. We do pharmacokinetic modeling with CompuCell. And I mentioned that we've done some modeling of diabetic retinopathy. But of course, diabetes is a very complex disease. And so there are almost a limitless number of diabetes questions one could ask. And so we'd be very open to ideas and suggestions about approaches to dealing with aspects of diabetes or doing better pharmacokinetic modeling. Jim Sluka, who is not, I think, on the call right at the moment, is an expert in pharmacokinetic modeling. And also Tarun, who is one of the participants in the class, is also an expert on pharmacokinetic modeling. Any other questions or comments before we break? There was a question, can you run the simulations out of the graphical interface running on a cluster?
Oh yeah. In fact, the grid that we surveyed and that James talked through with you yesterday about the drug therapy parameter variations, all of that was done on IU's supercomputing resources, specifically on carbonate. So that's all executed not within CompuCell itself, but actually in a general Python environment that calls CompuCell as a process. And in this case, lots of processes that are executed on lots of, cores in the clusters that we have available. So that's a generic capability. So that's something that is built into CompuCell and is documented to, varying degrees. But yes, in general, it is possible to do that. Otherwise, it would have taken about 3 years to do all of those simulations.
Okay, any other questions or comments before we break for lunch or whatever appropriate meal it is where your time zone is? I'd like to thank everybody for doing the mini presentations. I thought that went remarkably smoothly. And I do appreciate everyone condensing their ideas into such a short space. I'm sending out some links to help people organize. We'll post all of that information in the Slack. That Google Doc will continue to be active. And so we're looking forward to working with people. We'll do our best to help out, but it's really essential that people reach out. If you're not in a team and one of the talks took your fancy, please do reach out to the person who gave the talk directly and see if you can coordinate with them. And we'll have a session on Thursday where we'll investigate how people are doing in terms of organization. With that, I'd say we are done for today's module 2.2, and we will see you back in just under half an hour at 2:15 to begin understanding how to do chemical fields in CompuCell. Hello, everybody. We'll be starting again in just a minute or two. We'll wait for a few people to get back. This afternoon, Josua Aponte-Serrano will be presenting on chemical fields, which of course is an important part of any kind of multicellular model.
Hello, everyone. Do you all hear me fine? Yes. Okay. Good. Let's wait a couple of minutes to give everyone some time to reconvene. We'll wait until 2:20. And just as a reminder, this presentation will be recorded and will be live streamed on YouTube. And I will be back in at 2:20. Okay, let's get started. Again, as a reminder, this presentation will be recorded and live streamed on YouTube. My name is Joshua Aponte-Serrano. I am a PhD student at James Glazier's lab, and I will be talking about how to model chemical fields and diffusion using CompuCell today. Just as also as a reminder, you can find the slide deck I will be using is going to be, it's available in the Google Drive. And you can also find copies of all of the examples that I will be discussing today in the student demos folder in the same Google Drive as well. Just in case you fall behind, you can always refer back to the code. Okay, so this just for context, we are in this is module 2.3, chemical fields and gradients, the part 1. This is the second module of the day, and then I will be continue discussing chemical fields in the next module. So the first part of the in this module, I will be talking about what the biological ideas are behind chemical species, concentrations, and transport, and how this can be and how this can be thought of at least from a mathematical standpoint. Then we will continue talking about how we can then implement in CompuCell the concepts of diffusing and transporting molecules. And we will explore different conditions that lead to different profiles or concentrations of the different profile concentrations of these fields. Oh, by the way, I forgot to say, I'm gonna have to use my local version of CompuCell because I was having a little bit of problems running the demos in NanoHub, but I've uploaded the demos as zip files that you will be able to upload yourself and execute on NanoHub. I made sure that they were working, and they indeed are, but I was having a little bit of problems running it myself, so I will just use the local version. I'm gonna stop sharing for a second so I can share my full screen now. Tarun, I'm gonna have the split screen so I can both walk through the presentation and the code as well. So, so the basic idea of behind fields is that we want to model we want to be able to model the transport of molecules across or between cells or in tissue. And this is important, right? Because molecules, they are essential for the functioning of biological organisms. They control the transport of such molecules, control the important processes, including cell division, morphogenesis, development. They have roles in the transport of metabolic resources, of signals, and of other cellular products. So it's a fundamental component of the biology of cells and tissues that molecules are transported in or between them. So even though even though molecules are individual particles, we can make an approximation when we assume that these molecules are relatively high in that we have relatively many of these molecules, and instead of talking about individual particles, what we made an approximation and treat them as concentrations or densities, which means that we, instead of focusing on a particular molecule and the movement and motion of that molecule, we consider the number of particles that occupied a particular space or a particular area or volume, and area or volume depending on how we think about the domain of interest. and how the concentration of those molecules change in between different parts of that space. So again, the idea is if even though molecules, the interactions between molecules are occur at an individual level between one molecule and another, when we can make the approximation of treating them as densities, because we assume that there is enough of them such that we can treat them, we can focus on on the number of particles that occupy a certain volume or area on the space. So there's different transport mechanisms that lead to these particles or particles moving from one area or the other, or the concentrations changing in time. For the purpose of this workshop and in CompuCell, we focus mostly on diffusion as the main transport mechanism, although there are other mechanisms that are also important and play a role in transport of molecules in biological tissue, including advection and active transport. But again, the most important mechanism of transport that we will consider here is diffusion. And diffusion is a process in which molecules move from areas of high concentration to areas of low concentration due only to their random molecular motion. So this is important because because in principle, the diffusion does not require a external source of energy, except, let's say, the average temperature, right? But it doesn't require an external source of energy for the particles to, or the concentrations to change in time. So this is this is a main mechanism of transport that we will consider when modeling transport of particles in CompuCell. It's important to notice as well that diffusion, the concept of diffusion can be applied not just to individual particles, but it's an idea that it can be also applied to populations of bacteria, animals in the environment, heat, and different types of fluids. And although the underlying physics of all of these types of transport are different, the basic physics and the way to describe them are the same. So in cell biology, the diffusion is an important transport mechanism because precisely because it does not require cells to invest energy. On the other hand, diffusion is limited to transport over short distances, and this is and we will see this later on as we explore the mathematics. But it's important to, just as an idea, to understand that a diffusing molecule, the distance that it travels is proportional to the square root of time. So if it's just due to diffusion. So, for example, if a molecule takes 1 second to travel 1 micrometer, then it will take 4 seconds to travel 2 micrometers, and 100 seconds to travel 10 micrometers. So it becomes less and less efficient over longer distances. So again, that's the basic idea. So why do we? The idea is that we look at molecules. Molecules are important to model in a cellular and a tissue context, and we can make an approximation of treating them as concentrations when we assume that we have a sufficient number of them. And then what we care about is how the concentration changes from different regions in space. And we assume that these changes in concentrations are due to the diffusion of these particles, moving from higher areas of concentration to lower areas of concentration. Okay, so now how does the idea, now that we have that idea, biological background settled, let's see how CompuCell implements how these ideas are implemented mathematically and computationally in CompuCell. So let's just define the concentration at a particular voxel or pixel. Or as a number that determines, let's say at a pixel determines what the concentration is over that voxel or volume or area. So once we have defined that concentration, right, which means how many molecules occupy that volume or that space, then we can define the change in concentration as proportion as the using the diffusion equation, which has which is proportional to the gradient of the concentration, and the proportionality constant is what we call the diffusion coefficient, and this is going to be important. We're going to explore later on what the effect of the diffusion coefficient is on the transport and the change in concentration. But again, the idea is that if you look at a particular area of space, how the concentration in that space is going to change in time depends on the diffusion coefficient of the chemical that we're looking at or we're interested in, and the gradient of and the gradient of the concentration at that particular space. And it's important also to note that, and this is important, we'll see it later, that the net effect of diffusion is to erase the concentration differences across the domain. So for example, if we start with a, let's say we pour a drop of ink on water and we just let it settle there, we will, if we come back later, we would expect to see the ink to be uniformly mixed in the liquid, and that is due to the diffusion of the ink molecules. So what happens is that the differences in concentration when I put the drop in a particular domain were erased, were ended up uniformly distributed because of diffusion moving the molecules from higher areas of concentration to lower areas of concentration. And an important biological note here is that diffusion is not this if we look at a particular, let's say, tissue, we would not expect the diffusion constant to be the same everywhere in the tissue. Diffusion is faster in water than in the cytoplasm, because the cytoplasm it tends to be a much crowded space. It's usually faster in water than across a membrane because there's obstacles for the molecules to cross, and it's also faster in water than in other areas where the molecules might stick to. For example, if they stick to any components of the extracellular matrix, or if they bind to any let's say cell surface protein. So okay, now the idea, how do CompuCell represents this concentration? So if we imagine a two-dimensional lattice, the way CompuCell is going to represent the concentration at each particular site is going to be a scalar number. And that number determines what the concentration of the field or the chemical is at that particular location. And then the CompuCell diffusion solvers are gonna be in charge of solving the diffusion equation that I just showed previously to determine what the concentration, how the concentrations at each of the locations of the simulation domain changes in time. And there are multiple of these diffusion solvers in CC3D depending on the particular problem that of interest. Some of them might be more useful than others. I'm not gonna go into the details of this. If you are interested in learning about the different diffusion solvers, the documentation is here in the slide deck. We will focus on using the diffusion solver FE for the purpose of the examples in this module. So one last thing that it needs to be specified for solving the diffusion equation is what happens at the beginning and at the boundaries of the simulation domain. So at the boundaries, we have several options, and I'm explaining this conceptually now, but we will see examples of these in a minute. So there's basically three types of boundary conditions that we can have. We can have periodic boundary conditions, which is this example here in the middle. And in that case, we imagine that if a particle moves, let's say on the left edge of my simulation domain, it appears back on the right edge of my simulation domain. And we call it periodic because we imagine that the simulation domain that we're looking at is just a small, portion of a much larger simulation domain that are just copies of the same domain that I'm looking at. So that is why if one molecule disappears on my left side, one appears on my right side, because it's basically two copies of the same simulation domain, one side, one next to the other. Another option that we have is constant flux. And in that case, we can imagine that there are particles always coming in or out from the borders of my simulation domain. And our last one is a periodic boundary conditions. And what we imagine there is that there is always a source of molecules at our edges, that the value of the field at our edges is always the same. And then particles diffuse in or out based on the concentrations at those edges. And we will use this in the next couple of examples. It will become more clear as we walk through the examples. So let's just start with exploring what the effect of this boundary and initial conditions are. In this example, I will create a simulation with just one. I will show you how to create one field, how to set the diffusion coefficient that we already explained, and we will explore what happens when we start with different initial and boundary conditions. Yes, AJ, go ahead with your question.
My question was, if you consider the boundary more like a wall where the molecules bounce back.
There is an option, yes.
Yeah, what would be the appropriate boundary definition for the boundary condition in that context? So that it's not like it's fixed, it's not like it's wrapping around, but it's just simply like it bounced back. So that the boundary does have an effect. It causes things to accumulate somewhat over there.
Right. So as James and TJ are answering in the chat, we call that constant flux. Oh, constant flux set to 0 or 0 flux. Okay. So there is no flux across the boundary. That's basically setting a
when your
wall
boundary-
in which
when your
molecules bounce back. I'm sorry, go ahead.
When your boundary is a wall, should that not be no flux conditions?
Yeah, that's the zero flux or no flux condition, yes.
So no flux in your case is constant flux set to 0, is that right?
Yeah, so that's the way we implement it.
Okay.
Just one second, my earbuds disconnected and I can barely hear you, so just one second. So we call it no flux, right? Or zero flux or constant flux 0. But yes, that's the way we implement the basically the wall.
Perfect, thank you.
So with this example, what I want to show is what the effect of these different initial conditions are, and the boundary conditions, and then I'll show you also how to set how to set a chemical field and specify its boundary and initial conditions. So what we'll do first is we will create a new project. And we're gonna use XML, the Python X and XML. We can leave everything else. This part's general simulation properties have to do with what happens to cells. And since we're gonna be dealing just with fields, we don't really have to specify here anything in particular, except maybe the simulation domain. We can make it shorter just to make the simulation run faster. But we will have no cells, so we don't have to worry about specifying anything here. Same in the cell type specification. We're not adding cells at the moment, so we can hit continue. And then let's introduce one field in the chemical fields. Let's call it oxygen. And as I mentioned, we have different options of what the diffusion solver we want to use is. Those depends particularly on the problem of interest. For this, because we're going to be using relatively small diffusion coefficients, we can just stick to the regular diffusion solver. And then we hit add. And we don't need anything else. So we can hit continue. And then we will have one empty simulation that just has a chemical field. So let's walk forward. We add the chemical, we add the name. We can add the name of the field, select the diffusion solver, and we can hit add, and then we can add it. Pedro, in that particular case, right. And we can come back to that because this is an interesting example, and I can show there is an option for in CompuCell, for example, to set what the initial concentration of the field is, and then depending on what if you do not want your field to diffuse, for example, you can set the diffusion coefficient to 0, and the field won't diffuse, but you can still interact with the field depending on what your interactions of interest are. So coming back here, we add a field, we click next, and now we in the XML, we have the options to specify what the diffusion constant is. And remember, the diffusion constant is the proportionality constant that determines how the concentration changes in time at a particular rate. I'm sorry, one second. So the other parameters of the diffusion coefficient the other parameters of the diffusion equation, there's other parameters of the diffusion equation that we could also specify here in the XML, and we haven't discussed them yet, but we will introduce them later. But here, for example, you can also, there is also options to specify what the initial concentrations are based on either a formula that is dependent on X and Y refer to the positions in the plane, or you can import a file that specifies what the concentration is at each of the locations of the simulation domain. And then you can also specify what happens at the boundary. These are the options that I was talking about earlier. You have the periodic boundary conditions. You can also have a constant value at the boundary, or a constant flux or constant derivative. So, right, and depending on what our problem of interest is, we can specify our conditions accordingly. So now that we have created this simulation, let's just explore what happens if I start my simulation with a uniform concentration of 1 everywhere in my simulation domain. I am setting my decay constant to or I can set it to 0 because I am not using it at the moment. And then I'm gonna also specify periodic boundary conditions. So I'm gonna launch the poll just to make sure that everyone is if people are following. So all I've done is I created a project. I added a field that I called oxygen, and I didn't add anything else. And now I came to the XML where I can specify what the diffusion coefficient is. And I'm also specifying my initial concentration to be 1, which means that the concentration is going to be 1 everywhere in my simulation domain. And then this equation data we'll see later. So we don't need it at the moment. But then on the boundary conditions, I'm setting them to be periodic. So I'm gonna give everyone a little bit of time. This is important because we will be repeating this same exercise over and over of creating the field and coming and specifying what we want our diffusion parameters to be and the boundary conditions to be. Also, as a reminder, since this is exercise 2.31, I have an uploaded version of the file in the Google Doc, in the Google Drive. I would recommend having it open as a reference. Still work through the exercise, but if you have a question, you can always refer back to that document. Okay, so quick question. I am starting my simulation domain with my concentration being 1 everywhere. That's what I've set up in my initial concentration. In my initial concentration. or as my initial condition, and my boundary being periodic. What would you expect with, or what do you think it's going to happen once I run the simulation in terms of the concentration? Right. Nothing is going to happen. And why is that? Oh yeah, so that's the example. I'm sorry. I'll come if you ran my code has the next my code has the next part of this exercise also, that it initializes basically a drop in the middle of the simulation domain. So how do I boundary conditions? So if once you've added, you come to the XML file and you can find all of the chemical field information is always going to be contained in this table called diffusion solver FE. And then in that diffusion solver FE, you'll have the name of the field that you've just created. You'll have the diffusion data that contains the parameters, the diffusion parameters, the secretion data that we're not dealing with right now, so everything is commented out. And then you'll find the boundary conditions here. And then all I did was I commented out the constant values and set them periodic both on the X and Y axis. So, right, good point. So since we didn't create any cells, but once we create a simulation, the CompuCell it's going to load a steppable for initializing cells. So we need to get rid of that if we don't want any cells to be initialized in our simulation. So that's important. In this file as well, please comment out the uniform or blob initializer so we don't have any cells at the beginning of the simulation. Okay, so now what's going to happen, as some people commented out, is once I ran this simulation, I shouldn't expect anything to happen because diffusion depends on, right, is proportional to the gradient of the concentration. But if the concentration is uniform, there is no gradient. So there won't be any change in time of concentration in time. So that's what I should see. Another thing to point out is once I execute my simulation, CompuCell always executes, opens a cell field, opens a simulation in the cell field window. If we want to see the field, we come to this projection pull-down menu, and then we select the field of interest. And then I can continue running my simulation. And then as people commented out, commented, you shouldn't expect to see anything happening because we didn't have any, because the concentration is uniform across my simulation domain. Okay. So this was just, so that is in initial, so in diffusion data, you have the option to specify what your diffusion constants and decay constants are. And then you have additional options to specify the initial concentration. And you also have the option of uploading a file that has, let's say, what the concentration is at each domain of the simulation. What I did is that I set it, if you set it to 1, the initial concentration expression, that means that the concentration is gonna be 1 everywhere. Okay, so Now let's look at the next example. In the next example, we set the concentration to be, instead of 1, we're gonna set it to be this expression here. Well, it's 256 if your simulation domain is 256, but I set it to 100. So, because what we want is to place a dot in the middle of the simulation domain. Basically, we're simulating adding a drop of chemical on the surface of the simulation domain. Okay, I'm gonna share the poll again. It's 10 minus. So again, what I'm playing with here is the idea of starting with different initial conditions. In the first initial condition, I started with a uniform concentration across my simulation domain. Here, I am starting with a different concentration at the center of my simulation domain. Again, it's 256 if your simulation domain is 256, is 100 if your simulation domain is 100. You can always come back and check what the dimensions of your simulation domain are in this POTS data. Okay. So you can see the expression there. No, thank you. Thank you for the correction. So now, so quick question. Well, I mean, the answer is there already, but how do you, what do you expect to see, and what explains what you will see in the simulation, or what you see in the simulation? So what's happening? Someone can guess? So if I execute my simulation, right, basically the drop quickly diffuses and the concentrations diffuses across the simulation. So, okay, so I was going to explain this later, but I would just explain it now. So there is a way, if you're not seeing any changes in the field, you might need to adjust your viewing options. And I'll come back to this later. So, So, right. So Ben, I was having that same problem with Nano running this particular file in NanoHub, and that's why I'm running it locally. I'm not exactly sure if it's TJ might be able to help here, but I think it might be a configuration file that is really important. But I was also having that same problem that my mind was also crashing. So right, I start my simulation and I start with a dot in the center of my simulation. And as I run the simulation, I can see how the molecule diffuses away from, or the concentration of the field or the chemical is diffusing away from that original drop. The color bar, that is an excellent question. I will come back to this later, but the color bar automatically adjusts itself in the default options. So red is always gonna be the area with the higher concentration, and blue is always gonna be the area with the lower concentration. If I come to options here, I can fix what the color bar values are going to be. Let's set it to the minimum to be the minimum value being 0 and the maximum value being 10. And then I can see now that the values were readjusted and the heat map is not readjusting to the maximum and minimum. So if I start it over, I can see my dot at the beginning, but then it disappears because it's basically uniform. It has basically been uniformly diffused in the simulation domain. Okay. So again, I have copies of these files in my in the folder. In the folder, you can see in the file that I added to the folder, you will be able to see both exercises that I've done so far. If you comment out this line, you'll start with the uniform concentration. If you comment out this line, you'll start with the high concentration at the center of the simulation domain. Okay. So let's try now a little bit of a different exercise. So instead of having initial and boundary conditions, instead of playing with the initial conditions, let's now play with the boundary conditions. And what we will do is that we'll comment out the initial concentration expression. And if I comment out this, my simulation will start with 0, with the concentration of my field being 0 everywhere. And now what I want to do is that instead of having periodic and boundary conditions on the X and Y axis, I want to start with a constant value of 1 in both my X and Y axis. And let me explain what this syntax means. So constant value plane position min means the minimum on that plane. So in this case, 0. What is the value of the field at the boundary? 0. Constant max, plane position max, means the maximum on that plane. So in this particular case, 250 or 100, whatever the simulation domain was. Then same with the y-axis. I set the constant value to be 1 in the minimum and 1 in the maximum. Then if I restart my simulation, oh, so I was having this problem plenty of times. If you have different boundary conditions, it's gonna start with the last one you specified. So you have to make sure that you comment out the periodic boundary conditions such that when you execute the simulation, it only has the constant value. I kept having that error, running into that error earlier. So now my field is diffusing, but the reason I don't see it is because I played with I adjusted the maximum and the minimum of the fields. So I have to readjust it so I can actually see what's happening in my simulation domain. And I set it to 1 because 1 is the maximum at the edges. So again, I can rerun the simulation and you can see how the molecule diffuses inward from the boundary to the center of the simulation domain. Do notice though that in this particular case, the maximum value that the field is going to have, it's always gonna be 1, right? The value that I specified at the edge of the simulation or at the boundary of the simulation. I'm gonna stop here for a second and give everyone time to catch up. So if it's only diffusing from one side, make sure that you specify constant value min and max on both X and the Y planes. To get to get the simulation domain to look like mine, remember that you have to change from the self field to the oxygen. And in the settings, you need to let's just, for now, eliminate the fixed values of the field so you can actually see the fields. The concentration is gonna look different based on how you choose the heat map to look like, right? You can, here in this particular example, these are what we call floating. So the minimum and the maximum on the heat maps are always gonna readjust and adapt to the maximum and the minimum of the field in the simulation domain. If instead I want them to be fixed, I can go to settings and fix them such that the colors are fixed to the values that I specified. So Tony, just again, make sure that you have specified values of the planes in both X and Y axes. And just so everyone remember, I have a copy of this particular file for reference. And it has all the options that I've played with. I've just been commenting stuff in and out so you can all see the different condition sites I've specified. Right. So you need to make sure that you're adjusting the constant value on both X and Y axis and not the constant derivative. Right? Because remember, constant value is one type of boundary conditions where the value of the field is always constant at the boundary, whereas constant derivative is a different condition that we will see next. Yeah, I'm gonna give people some time because this is the end of exercise 2.31, and I want everyone to at least get some familiarity with it. So again, just as a reminder, once we've specified the field in CompuCell and we open the XML, we will see this diffusion solver steppable that will contain the data of it's going to contain all the information pertaining the chemical field. The first part is you're gonna specify what the diffusion coefficient, the diffusion data is, the diffusion coefficient, the decay, and the initial conditions. And then you have another section where you just specify what the boundary conditions are. Right, I commented it out for this particular simulation, for the simulation where I'm starting with a constant value at the boundaries. Cato, do you have a question? How to fix the lookup table? One second. There you go. So that is the link to these files. And you can, the exercise I'm looking at is the 231, exercise 231. So again, I'm giving everyone time because all the other exercises in this and the next one depend on we understanding how to specify our diffusion data in the XML. Yes. So again, I'm gonna redo everything I did quickly because I can comment out stuff here. So the first simulation I started with so the first thing I did after loading the XML was that I set the decay constant to 0 because we're not going to worry about decay right now. We just want a diffusing field. And the first simulation that I did, I specified my conditions to be periodic at the boundary and my initial concentration to be 1 everywhere. And when I do that, what I see is a uniform field that nothing happens because there is no gradient in my simulation. Okay. The second example, instead of starting with a uniform concentration everywhere, I started with a dot or a drop in the middle of the simulation domain. And then what I see as I run the simulation is how that drop diffuses from the center of the simulation domain, where I placed it at the beginning. Okay. The next example that I showed was what happens if instead of having some initial concentration inside the simulation domain, I set a constant value at my boundary. And I noticed people were having this problem. I have that problem all the time too, that I forget that it has to be, the tag in the XML has to be constant value. If I specify constant derivative, that is gonna do something different. So instead of having an initial concentration, I started with some value, some constant value at the boundary. And in that case, I can see the field diffusing from the boundary inwards. Yes, Ford. That is, to do that, to readjust the color schemes, you can come to settings, and then it's gonna allow you to adjust what your maximum value and your maximum minimum is gonna be. And remember, your maximum value is always gonna be red, and the minimum is always gonna be blue. Okay, and then the last, and now I'm going to continue with the last part of the exercise, which instead of having constant value, we're going to have different boundary conditions with the constant flux or constant derivative at the boundaries. And then when I run that simulation, I can again see the chemical diffusing inwards from the edge of the simulation. But now we can notice that the maximum of the field, instead of being 1, as in the previous example, it's always increasing. Anyone has a guess as to why is that the case? Right, because constant derivative is the same as a constant flux. Dan, that's an excellent question, and the answer is the latest. What the numbers here mean are gonna depend on how you're interpreting the values of the field. And generally, you're free to interpret them as you want in terms of let's say, as a concentration, number of particles, and so on. But yeah, you have to be consistent once you've said, for example, what the length of your pixel is. Okay, so yeah, and so I'm gonna answer the question here. The reason we are seeing this number continue to increase as opposed as in the previous case where it was constant, is because in this case we're constantly supplying to the simulation domain from the edges, right? So the concentration is always gonna keep increasing because we're basically always supplying from the edge. Whereas in the previous one, instead of supplying, we had just fixed the value at the edge and let the molecules diffuse inward. But in this particular case, we are sort of like supplying in from the edges. So that's why it keeps increasing. And that is the difference between having a constant flux and a constant value at the edge of your simulation domain. Okay. So now that we have explored our different options of initial and boundary conditions, let's look at what happens if we try to solve we solve the diffusion equation with a fixed boundary values and no decay. So in that particular case, we can solve the diffusion equation. This is a diffusion equation quite easily. And the solution of the diffusion equation is gonna be this. It's basically saying that the concentration is gonna change linearly on the position. And that the equilibrium concentration is independent of the diffusion coefficient. So for example, if we start at one boundary with a value of 100, then the diffusion coefficient, the solution to the diffusion equation with no decay is gonna be this equation here. Of course, when we start the simulation, depending on the initial conditions, we will not necessarily reach this equilibrium concentration. But if we let the system run for long enough, the system will eventually equilibrate to the solution of to the solution of the diffusion equation. But the time that takes for the system to equilibrate to that solution depends on the diffusion coefficient. And that introduces the idea of the diffusion time. And the diffusion time is usually the time that it takes a molecule to travel a distance at a distance, let's say L. So you can see that the and this is what I was explaining earlier, the time that it takes increases as the square of the distance. So if it takes 1 second to travel 1 micrometer, it's gonna take 2 seconds to travel it's gonna take 4 seconds to travel 2 micrometers. It's gonna take 100 seconds to travel 10 micrometers. So diffusion is effective over short distances, but it slows rapidly over long distances. And that is why, for example, cells take advantage of diffusion over short ranges where they don't have to invest energy. But if they want to transport molecules across bigger distances, they generally have to rely on active methods of transport that require using energy. So now what we will do is that we will specify, we will see how we can implement this linear gradient or this linear concentration using CompuCell and playing just with our boundary conditions. And what we will do is that we were gonna write a simulation where we imposed a constant concentration on the left side and a 0 concentration on the right side. And in this particular example, we're also gonna learn how to measure that gradient and how to plot it. So when we create this simulation, we have to remember that we want the diffusion constant to be uniform everywhere. So we won't have cells. We want no decay. And we want and we also want fixed value on let's say the X and Y axis, on the X axis, and then boundary conditions on the Y axis. So let's try to get that, let's try to generate that simulation ourselves. So we come back to, I'm gonna relaunch the poll. Okay, so we start a new project. Oh, and just for reference, again, this is example 2.3.2. But if we want to start from the beginning, we create a new simulation. I'm going to call it Field 2. And I'm going to use, just because it's easier for me to think in powers of 10, I'm going to use a simulation domain of 100 by 100. I'm going to have no cell, and I'm going to add an oxygen field. And then I finish with my simulation. And then I can come to my XML. And again, I have to specify the properties of the chemical field in this diffusion steppable. So let's set the global diffusion coefficient to be 1. We are not interested in our molecules decaying at this moment. We'll come back to that idea later. And then I said that we want in the x-axis, we want our value to be 10 on the minimum and 0 on the maximum. And then on the y-plane, we want our conditions to be periodic. And again, remember, every time we create a simulation, but we do not want to have cells, we have to delete the initializer. Okay. And that's all I need for this simulation to run. I want you to give it a try and let me know if it's running on your case and what you see when you run it before I move on. Seems like most people caught up with this exercise. So can someone tell me what they're right? So now you basically start with red on the left side and or the chemical on the left side, and it moves to the right side. So I'm gonna execute the. Remember, to see the field, I always have to come to this pull down menu, and then I can execute the simulation. And I can see again, so for example, here, I can see that the numbers are, that the heat map is, it's free floating because the minimum and the max, the minimum and the maximum are adjusting themselves. So if I wanna fix them, I can come to my settings and I know what my minimum, my maximum is, right? Because it's coming from my left edge, and that is 0. And I know what my minimum is because it's the value that I established at the right edge, and that is 0. It's the maximum is 10, the minimum is 0. So I can adjust that, and I should be able to see my gradient uniform or linearly distributed in my domain. Okay. So this is what I was explaining earlier. If you want to adjust what you see, you can do it in settings. So for example, if I set my maximum to be 1, then my field is gonna look different because everything that has a value of 1 or bigger is gonna be printed red. So that's why it's correctly interpret visually this simulations, you need to be very aware of what your display settings is. And there's another visualization option that we have where we can show lines. So if we go to settings and we click on show the scalar contours, this is gonna draw lines that show regions of the domain that have the same concentration. So make sure that your simulation is running. To make sure that you can, you need your simulation to be running in order for it to adapt to be able to fit, to play with the settings, visualization settings. So again, these contour lines show regions of my simulation domain that have the same value of the concentration of the field. And I can play with how many of these lines I want to show. Okay, so we call this linear gradient, right? Because the field is linear, it decreases linearly from one edge of the simulation to the other edge. And that's why these contour lines are equally spaced between each other. Now, we can also Yes, absolutely, Tony. So all I did in the code was I set my decay constant to 0. I set in the boundary conditions to be constant value on the X plane, 10 in the minimum, 0 in the maximum. And periodic on the y-axis. Okay, and I'm gonna show you now how we can measure and plot the concentration, not just look at it. I'm not necessarily sure if we cover plotting already. This is gonna be a refresher. If not, this is probably the first time we'll see plots, but this is also very important. For plotting, we always do our plots in the steppable in our Python steppable file. We've been so far playing mostly with the XML, but if we want to add scientific plots to our simulation, we do that in our steppable files. Once we are in our steppable files, if we want to have access to if we want to have access to the code that would allow you to generate these plots, we come to this pull down CC3D Python pull down menu. And we come to scientific plots. And there's a couple of options here. We have a setup option and an add data point option. And then we have this erase plot option, which we will be using. So if the first thing we need to do is to set up our scientific plot window. And we do that by generating this code snippet inside the start function of our steppable. When we click on it, we get this code. The first part of the first line of the code basically creates the plot window, and it has as properties a title, an x-axis title, and y-axis title, and then whether we want our x and y scales to be linear or log scales. We talked about that yesterday. So what we will do is that we will call our plot, we will call it concentration versus or per position. Our X title is gonna be the position, and our Y axis title is gonna be concentration, and we want them to be linear. And then the next two parts of the code snippet create some data series that are gonna be added to this plot window. Since we just want to measure the concentration, we just want one data series, and let's call it CON. And we can specify what style we want. In this case, I'm gonna have dots, but it can also be a line, and the color and the size of the markers. And if I execute my simulation now, I'm sorry. If I execute my simulation now, I can see that I now have a concentration per position window and a plot. But as I execute the simulation, I don't get anything, nothing is being plotted because I haven't added any data to the data series. So to add data to the data series, I can come back again to the CC3D Python scientific plots, and I can add data point, and this is going to be inside my step function. And the code snippets suggests this syntax. It's important to match the name of the data series to the one I created in the start function, right? And then the second argument is going to be what is going to be plotted on the X position, and the Y, it's going to be what is going to be plotted on the Y position. So if I want to measure and plot the concentration, conceptually what I have to do is I have to iterate through the X, let's say on the X plane, and measure what the concentration of the field is at that particular location, and then plot that location and the concentration. So let me just relaunch the plot to make sure that everyone is following. Okay, good. Okay, good. So the way I do that is I'm gonna iterate, I'm gonna create a loop where I iterate over my X domain, and I do that. I have this property called self.dim.x that gives me back the length of my X domain. And then I can add this inside of, I can move this add data point inside of this loop. But now I need to basically get or fetch what the value of the concentration field is at that particular location. And to do that, I have this, I can come back to my CC3D Python pull-down menu, which is gonna be your best friend. And I come to chemical field manipulation. And I need two things. I need a reference to the field, get field reference. And I also need to get the field value. So now that I have those two things, I can, so I know I have 1 more minute left for this module. I'm gonna run over until 3:50, so I can finish showing how to plot, because this is important, how to measure the concentrations, because this is important. And then we'll continue with the next module after. So for referencing the field, all I need is to tell that field that, and then I have to change to the actual field name as created on the XML, which in this case is oxygen. And then to fetch the value, I just use this syntax where I specified the field position that I'm of interest. So what I can do is I can move this inside of the for loop. And then ask the question, sorry. And then I can ask the question, what is the value of the field at position X? And I also have to specify Y. It's not important in this particular simulation because Y is the same value across the Y domain. But if it wasn't, I would have to specify what position in the Y domain I'm also measuring my concentration. And in this particular case, I am measuring halfway of the simulation domain. So now I can pass on this value to the add data cities. And then because I want the plot window to show how the concentration changes in time, I need to, after I plot, I need to erase the data and then basically plot the concentration again. And the way I do this is I can come to CC3D Python scientific plots and erase plot, and then it's gonna, that's also added in the plot function, in the step function, and make sure to add it before your for loop. So this is important because we will also come back to this in all the other examples. And now if I run my simulation, I can both see how my chemical is diffusing in the simulation domain, but I can also see how the concentration is changing at each of the locations of the domain. And at equilibrium, I should expect this to look like what? Since it's called a linear gradient. So it's supposed to look like a line, right? Because and that is why we call it a linear gradient, because the concentration is decreasing linearly from the source to the sink. Since we ran out of time, what I wanted to show next was what happens if we, and we will come back to this, but what happens if we, and I'm gonna start this next model here because it's important, but just as a teaser, what happens if we change the decay that we had specified it to be 0, if we instead change it to be, let's say, a non-zero value, then the profile of my concentration changes, and instead of decreasing linearly, it now decreases exponentially. And we will start the next module with this example, but this is just as a teaser, because I wanted to show you what the difference between having a linear gradient is at a linear and an exponential gradient is. And we can see that also how long does it take to reach that equilibrium value depends on the value of the diffusion coefficient. Yes, the PI file it should look like this. And this is important because we will continue using this code, this part of the code for all the next couple of examples. And I'm gonna stop here and I will come back and start over in this part of the module. In 10 minutes. Please do feel free to ask questions while in the meantime, if you need some time to catch up. Oh, okay. I'm gonna hold on. I'm gonna share my screen again, my full screen, so everyone can see what the plotting looks like. But again, remember that all of these options are available for, you don't have to know them from the top of your head. You can always reference them from the CC3D Python pull-down menu. Remember, we use scientific plots, the setup, the add data points, and the erase plot. And we also use the chemical field manipulation. We use the get field reference and the get field value. And the reason I need both is because I need my Python code to be able to reference back my XML code. And that I do by getting this get field reference.
You're muted.
You're muted, Josua.
Oh, I'm sorry. I'm sorry. I'm gonna keep up my code in case people want to finish. I'm gonna in case people want to finish coding their plots while I load the slides for the next module. Okay, we will give everyone 2 more minutes to reconvene, and we'll start our next module. Okay, welcome back, everyone. We will continue discussing how to model diffusion and implement chemical fields in CompuCell. This is now module 2.4, although we will continue with some of the exercises from the previous module. And just as a reminder, this presentation is being recorded and live streamed. So in the previous module, we talked about how to model chemical concentrations and diffusion of these chemicals using CompuCell. In this module, we will discuss how to add cells and what cells can do to these fields. They can either act as membranes, or they can secrete to and absorb from this module, from this chemical field. But before getting into that, I wanted to finish the last part of the previous module because it is important. The last part of the module we saw was the implementation of the linear gradient, right? And the way we implemented that linear gradient was by establishing a fixed value of the field in one edge of the domain and another value at the other edge of the domain, and having the molecule diffuse between one edge and the other. But in our simulations, the molecules diffuse freely without being interrupted. In reality, in biological context, and what we see is that as we implement that field and we measure in, our simulation starts with all the concentration of the chemical at one edge, but then that concentration diffuses through the domain until a equilibrium concentration is reached, where the concentration decreases linearly from one edge of the domain to the other. And this linear concentration, again, is due to the fact that the molecules are diffusing freely from one edge to the other. As a bonus, I have it in the code that I shared, the 2.3.2 code. You can also, since we solved the diffusion equation under those particular conditions, we know what the equilibrium concentration should be of the field, and then we can plot the actual concentration versus what the equilibrium concentration would be. And if we wanted to measure, for example, the time that it takes for the concentration to reach equilibrium, we could do so by, let's say, subtracting and measuring how fast the concentration profile reaches the equilibrium concentration. But that is as a bonus that I left in the code that I shared with you. Now moving on to solving the diffusion equation with decay, we need to take into consideration that in living organisms, molecules do not freely diffuse across tissues, right? They interact with other molecules that are consumed by, let's say, cells, or they undergo chemical transformation because of chemical interactions with other molecules. molecules. And the way we model the fact that this type of, that the molecules can be consumed, or that they basically, they are basically used up, is by including a decay term in our diffusion equation. And it's also important to know that this decay is gonna determine an important, very important biological quantity that we call the diffusion length. And the diffusion length is basically the typical distance that a molecule is able to diffuse in a particular tissue, taking into consideration that the molecule is both diffusing but also decaying. And that diffusion length depends on the particular molecule that we're talking about and the particular tissue that we're talking about. So for an example of this decay is oxygen diffusing from the capillaries into the tissue. It has a characteristic length that the oxygen molecule can diffuse into the tissue, and this determines, usually determines, for example, the spacing, the minimum spacing necessary between two capillaries to provide appropriate oxygenation to the tissue. And so on. This diffusion length determines important time and length scales for different biological processes. So as I said, the way we can model this consumption of molecules or this exhaustion of molecules is by, or the decay of molecules is by including a decay term in our diffusion equation. So remember, our diffusion equation without decay was this first part of the equation. And now we add an additional term that is accounts for the fact that our molecules decay in the domain as well. And then it's important to notice that the decay is going to be proportional to the concentration. So the more concentration, so the initial concentration or the available concentration, and that is going to be, and then how fast it decays is going to be determined by what we call the decay constant. If we solve this equation, what we see is that instead, as in the previous case where the concentration profile changed linearly from the source, the concentration in this case with the decay term is going to decay exponentially from the source. And the exponential rate of decay is going to be determined by both the diffusion coefficient and the decay rate. And then, such that the diffusion length, now we can define it mathematically, is the distance over which the concentration decreases by a factor of E. And that is usually so we can determine what the diffusion length of a molecule is by taking the square root of the diffusion coefficient over the decay rate. Biological molecules have, depending on the molecule, have typical diffusion lengths of 5 micrometers to 150 micrometers. So if we come back to the previous simulation that we were exploring, we can come back to the code that I shared in exercise 233. I'm gonna just load that one. So I would recommend everyone to do the same, to load the code that I shared, exercise 233. And some of this is gonna be familiar to us already. We can see that I'm plotting the concentration of the field at the by measuring the concentration across my simulation domain. And in the XML, I can see, Juliano, can you please share the link to the folder where the demos are? If I come back to the XML, I should be able to see that I've specified the boundary conditions that I needed, the same boundary conditions as in the previous example, periodic on X on the Y plane and fixed value on the X plane or X axis. But now I am, instead of setting my global decay constant to 0, I am setting it to 0.01 or 0.1. And when I run that simulation, what I see is that instead of decreasing linearly, my chemical, the concentration profile decays exponentially from the source. So we can set the diffusion. I showed the example where I set the diffusion, the decay constant directly into my XML, but I can also, for those of you who have been able to use it, I can also do it using my model editor. If I go to the steppable tab, the diffusion field tab, and the diffusion data tab, now I have the global diffusion coefficient and the global decay constant. If I set that decay constant to 0, I am again in the domain where I have a linear profile, concentration profile. And again, how fast does it take to equilibrate depends on my diffusion coefficient. But if I set my global decay to a non-zero value, then my profile is gonna decay exponentially instead. Okay. And let me share the poll to make sure that everyone is with me. And then you can play with this simulation yourself. You can see, okay, what happens if my decay constant is small, then the smaller my decay constant is, the closer my concentration profile is to the linear case. And then the bigger it is, the faster the exponential decay of my concentration profile. And then again, how fast does it take to get to the equilibrium concentration? It's gonna depend on the diffusion coefficient. And note that, of course, because the diffusion length is determined both by the diffusion coefficient and the decay coefficient, when my decay coefficient is not 0, the profile is also, the equilibrium profile is also determined by what my global diffusion coefficient is. Stephen, I can show my code again. It's just the same code as before, except that we've set the global decay constant to a no zero value. In this particular example, I set it to 0.1. Okay, cool. So, so I'm gonna leave this code to you to play to get some familiarity with the idea. Again, if you're able to use a model editor, I would recommend you playing with these values and understand how they control what the profile, look at two things, right? What the equilibrium concentration is, and then how fast it takes for the profile, for the concentration to reach that equilibrium state. So I'm gonna show the code for those of you who need a little bit more of time. I'm gonna give you, if you checked on needing more time but finish the exercise, please do check on yes, so I can get a sense of how many people are finishing the exercise.
People can't change their vote, so we have to relaunch the poll.
Oh, so okay. Well, so okay, good. So I'm gonna yeah, I'm gonna assume most people have finished already. So, but again, you can build on the previous code. Okay, good. Yeah. So I will continue on then. So as a bonus, I added additional code in the code that I shared with you. I added a line that shows what the value is when the original concentration decays by a factor of E, right? And I added a line that measures at what distance that concentration has decayed by a factor of E, which is gonna basically determine what your diffusion diffusion length is. And notice that this is just for the particular example where my diffusion coefficient is 1 and my decay constant is e to the -1. If I change those, then I would have to come to this code and readjust those values to see what the actual diffusion length is for different values of those parameters. Okay, so let's continue then with the material for module 2.4. This is the second part of the chemical fields presentation. And here I will be discussing how to use cells to specify diffusion coefficients that vary at different locations of the simulation domain. So in our previous examples, we assumed that the diffusion coefficient was uniform in all of our simulation domain, but we are also able to set varying or local diffusion coefficients, and we do that by introducing cells into our simulation domain. Then I will show another way cells can interact with the field is by secreting some of the chemicals to the field, and also by absorbing from those fields. And I'm gonna show you examples of how those can be implemented and how they affect the concentration profile. So again, in real biology, the particles do not diffuse equally across the same let's say, biological domain or the same space, because the space is usually occupied by different let's say there's areas where there's higher density of certain types of molecules, or there is actual barriers that prevent cells from moving through certain areas, or cells in it or molecules interact more or less with other molecules at different regions of the space. So that will effectively, if from the standpoint of modeling transport as diffusion, that would effectively be modeled as a diffusion coefficient that varies spatially. So in terms of how the diffusion equation is how we can mathematically represent that in the diffusion equation, in this particular case, our diffusion constant is now a function of the particular location I'm looking at. The diffusion equation becomes a little bit more complicated, but we can neglect this term if we this third term if we assume that the differences in concentrations, if the differences or the changes in the diffusion coefficient are not too big. So we'll stay with this last part of the diffusion equation, which basically states that the diffusion coefficient, the change in concentration depends on what the value of the diffusion coefficient is at that particular location and the gradient of the concentration. And as I said, the way we implement this in CompuCell is by specifying cells and then specifying what the diffusion coefficient is inside of those cells. So to exemplify this, we will continue building up on what the examples we were showing earlier, but we will introduce cells that will basically function as a membrane the chemical can diffuse through. So the idea is that, again, we will create a simulation with a single field and a single frozen cell type, and then we will use a uniform initializer to draw a membrane in the middle of the simulation domain. And Juliano earlier today showed a bit of how to use a uniform initializer to draw cells. And then we will set a different constant local diffusion coefficient inside the cell, the membrane cell type, to make the membrane semi-permeable or to make the membrane impermeable. And then as a bonus, we will open a channel through this membrane and look at how diffusion changes if it's diffusing through a channel in the membrane. So let's do this together. We will create a new simulation. I'm gonna call it membrane. And again, I'm gonna do it, although it says 200 there, the examples are 100 by 100. So I'm gonna type it here. It's 100. Now I am actually gonna be using cells, but since they are gonna be frozen, they're not gonna be moving. I don't necessarily care about what happens at the boundaries because my cells are not gonna be crossing any boundaries because I'm gonna freeze them. Now I do have to introduce a cell type, and I'm gonna call it membrane. And here I can check to have our cells frozen. And that, again, that means our cells are not gonna move. Let me make sure that my chat is. I continue, and then I can add. Yes, I can go back here to the cell type specification.
Another one.
Okay, to the simulation properties. So all I did was I specified a 100 by 100 simulation domain. And then I added a membrane cell type. And it's frozen. And then now I'm gonna add an oxygen chemical field. Again, I keep calling this oxygen, but you can call it as you please, and depending on what molecules of interest you model. And then although I have cells, since they are frozen, I do not have to activate any of the cell properties or behaviors because my cells are basically they're gonna be placed there, but they're not gonna be moving or doing anything except serving as a barrier for diffusion. Then I can finish my simulation. I can finish my I can finish the wizard, and then I can come to my XML file. First, I check that I've added my cell type membrane, that it's frozen, and this tag indicates that my cell is frozen. And now I can see that I have additional information in my diffusion solver stepable. I have the global and diffusion parameters, but I also have the diffusion and decay parameters that are specified by cell. And this is where I can change what the diffusion is inside of cells belonging to this membrane cell type. For this particular simulation, I want no decay. So I'm gonna set my global decay constant to 0. And I'm gonna, and my local, or my decay coefficient inside the cell, I'm gonna also set it to 0. No, sorry. And the diffusion inside my cell, I'm gonna make it 1e to the minus 5. Okay, and I want the linear gradient again. So what I'll do is I'll set the value, a constant value of 10 in the minimum of the y-axis and 0 on the maximum of the y-axis. And I want periodic boundary conditions on the y-axis. To initialize my cell membrane, I use this uniform initializer. And what I'll do is I'll set it basically in the middle of my X domain, going from 45 to 55, all the way from the bottom of my Y domain to the top of my Y domain. Okay. So I'm gonna pull and relaunch just to make sure that everyone's caught up, because this sets up everything for this particular exercise. So again, I have a global diffusion coefficient that I set to, let's set it to, actually to 1. A global decay constant that I'm setting to 0 because I want the linear gradient. And then I am making the diffusion coefficient inside my membrane to be 1e to the minus 5. Decay inside to be 0. And then I set the linear gradient by having constant values on the x-axis, periodic on the y-axis. And then I initialize my membrane by specifying the regions in which my cells are gonna be created. It's gonna be from X position 45 to X position 55. So that's basically the center. And in Y to 0, from 0 to 100. Now, if I execute that simulation, first, I should see the membrane in the middle of my domain. And then I'm gonna open another window so I can see the field. And I want someone to execute them on their end and tell me what they see before I run it on mine. Right. So, but what happens when it reaches the cells? What happens to the fields when it reaches the cells? Right. It accumulates. And that is because it's the membrane I created is semi-permeable. So even though the chemical can diffuse through my membrane, it diffuses slower. So you would see that the chemical field can get through that membrane. And I'm gonna execute it now. Sorry, I'm sorry, I forgot. There is this, once you created a cell field, the steppable automatically sets your cell type to secrete to that field. And we don't want that right now. So everyone will need to come back to their XML code and comment this in secretion data. Please comment out this secretion tag. So actually in this particular case, to comment, I set, right, you can set secretion to 0 or you can comment it out. Those are equivalent. If you want commented out, you place your cursor on the tag, then you can come to file, to edit, and you can save, and you can hit in block comment. So in this particular example, the diffusion coefficient is so small inside my cells that basically the chemical field is unable to diffuse through it. So let's increase the diffusion coefficient to be 0.01 instead. And you see that it's still diffusing through the cell field, but very, very slowly. Now let's set it to a 10-fold slower or 10-fold smaller diffusion coefficient. And now you can see that that it's coming through the membrane, but when it gets to the membrane, it slows down. Once it's past the membrane, it diffuses again faster as before crossing the membrane. So if you want to slow it down, you basically decrease the diffusion coefficient on the cell type membrane. So in this case, this is a semi-permeable membrane, right? The chemical diffuses through the membrane, but it diffuses slower than in the outside. If I make it very small, then I make my diffusion, then I'm effectively making my membrane impermeable. So it's coming through this edge, but it cannot cross this membrane. Joseph, make sure that your field properties are fixed. Again, because the visualization of the field is going to be dependent on what your visualization options are. So in my particular case, I have them fixed from 0 to 10. It also depends on what your diffusion constant might be. If your diffusion constant is very fast, then it's going to fill in the space. So that is an excellent question, Farnoosh. Yes, that is an option. That is an option we can we can make ourselves consume from the field. We also have other more active ways of doing it with where you have more options as to specify how the cells consume from the field. And I'll have examples after this one. But yes, that's an option. Okay, so let me, relaunch and make sure that everyone's with me. So again, I can always play with I can always play with the parameters of my diffusion by coming to the model editor. And then now I have both the global and global diffusion and decay coefficients, but I also have the local decay and diffusion coefficient. So I can set it, let's say, to the same as the field, as outside of the membrane, in which case the chemical acts as if there were no cells in the simulation domain. Or I can decrease the diffusion through the membrane, or I can make it impermeable by setting the diffusion to 0 in my cells. So in this particular case, since I already had some chemical inside the membrane, when I change it to 0, that chemical is just going to remain there. That's it. Aaron, that is an excellent question, and I'm actually not sure. Maybe TJ or Juliano can probably answer that question better. TJ, do you know?
So the global decay is applied everywhere that does not have any other specification. So if you specify a decay coefficient for a particular cell type, that specification for that type will override or supersede the global decay constant, and that's also true for the diffusion coefficient. So the global values are the de facto value for everything that doesn't have any other specification.
Yeah, so it's not additive.
Correct.
Okay, so most people are caught up with me here. So I'm gonna continue because the next parts are also important, but just as a right, this is exactly what I showed already. Oh, I can also plot, right, add the plots to see what how the concentration is changing. The plot is the plot code. This is why I wanted to spend some time of it on the last section, because it's basically universal, so we can just copy it and paste from the previous simulation. And I don't need this, but I need the basic one, but I'm measuring the concentration. I'm creating the plot window, and then I'm measuring the concentration. And if I just copy and paste that into this, then I can see into this simulation, then I can see into the step hours of this simulation, then I get my code. And this is what we'll do, because we'll get the plots for all of the next couple of simulations. Okay. Good. So, and as a reminder, you can find examples of this code in this folder in the Google Drive, and this will be exercise 2.1.241. And then you can play with the permeability of the membrane. And this is just as a teaser. I'm gonna leave it as a homework, but we can, by playing with the initializer, we can open a channel in our membrane and then see how that changes the diffusion of our chemical. And these are examples of diffusion through, in this particular case, the membrane is impermeable, but I open a channel that it's that lets the chemical diffuse through. So important here is that as it approaches, remember that the contour plots show the regions of the domain where the chemical concentration is the same. And you can see that inside the channel, the chemical basically diffuses more or less linearly. And then outside, it has this curved shape until it's far away where it diffuses linearly again. But this code is also available for you in that 2.4.1. It's just commented out for the purpose of this exercise. But I'm gonna leave it as an exercise to you to play with the width of this channel and see how that changes the diffusion of the chemical. So another way cells can interact with the chemical fields is by secreting molecules to that chemical field. Cells do secrete biological molecules. They have different ways of secreting these molecules. From our standpoint, we neglect the details of the specific secretion mechanism, but the main idea or the biological idea the main idea behind the secretion is that the net effect of the cell secreting particles is that the concentration above the, let's say, apical surface of the cell the concentration of that chemical increases as the cell secretes more and more of that chemical. From the modeling perspective, we can model secretion by adding an additional term to our diffusion equation, and this is what we call a source term. And in particular, the source term is gonna be dependent on the cell that occupies that particular location. So now our diffusion equation is going to be, so the concentration at a particular location is gonna be determined by the gradient, the diffusion coefficient, the decay, but also how much has been secreted into that particular domain by the cells that are, let's say, directly beneath it. So CompuCell has many different ways in which we can implement secretion. So the idea again is that we have a special plugin that allows that facilitates or enable our cells to secrete an uptake from a particular field, and then we specify the rate or the specific format or the form that we want our secretion to have. There's two ways we can specify secretion in CompuCell. We can either do it directly in the XML, or though our options are a bit limited in that particular case. Or we can do it in the Python steppable, which gives us much more control over the type of secretion that we want to have. And this is going to be the same for uptake of absorption. So if some of the options that we have to implement secretion is that we can secrete at the boundary of the cell, inside the cell at the boundary. We can also have secretion happen at the boundary of the cell, but instead of inside the cell, at the outside of the cell. And we can have secretion also happen inside the cell over every pixel that the cell occupies, or we can have secretion happen at the center of mass of the cell. Another option that we have that is commonly used, especially in some of our modeling papers, is that we can have medium become a source of the chemical by having medium secrete. Even though medium is not a particular cell type, it's not a cell type that has the properties of other types of cells in CompuCell. Secretion is something that our medium can actually do. And the advantage of having secretion done by medium is that we can control much easier what the equilibrium concentration of the chemical is in our simulation domain. And this is what we're gonna do now. So mathematically, if we think about it from a mathematical standpoint, the equilibrium, what the equilibrium concentration is, once we have uptake and decay, it's going to be the ratio of the uptake rate over the decay rate. And to prove that, we will create a simulation domain. We will add an oxygen field, and then we will enable the secretion plugin and specify the secretion rate of medium to be some particular value, and see what the equilibrium concentration is that we reach in the simulation domain. So let's do that together. I'm gonna create a new simulation. I'm gonna call it secreting medium. Again, I'm gonna make it 100 by 100. I won't have cells in this particular simulation, so I don't care about the details, the other details. Again, I have no cells in this particular simulation. I add the oxygen field. And now here, I do have to enable the secretion plugin. And then once I enable my secretion plugin, I get this additional tab. Usually by hitting continue in this previous section, I will complete no cells. I will complete my simulation specifications. But now that I have enabled the secretion plugin, that allows me to specify the that it gives me this additional tab to specify the secretion options. And what I'll do is I'll specify the secretion of oxygen by medium as a uniform secretion at a uniform constant rate of 0.2. I add as an entry. And then I finish my simulation. So now I can come to my XML. Let me launch this. And then in my XML, we're going to use this as a template. So what I'll do is as we can notice, since we enabled the plugin step, the secretion plugin, now I have this additional plugin information here in the XML. But that same information I can also specify in the secretion data in the diffusion solver step. And I prefer doing it there because that way I ensure that the secretion and diffusion are implemented at the same time, rather than one being implemented before the other one. So what I'll do in this particular case is that I'm gonna delete the information in this secretion plugin, but not the plugin itself. Just gonna delete this information. And I'm gonna specify it again here in the secretion data. Before deleting, just copy this secretion information. Paste it in the secretion data in the steppable, inside the steppable, and then now you can comment out or delete the specifications inside the secretion plugin. Okay, now I'm gonna specify my diffusion constant to be 0.01 and my decay constant to be 0.001. So yeah, in the secretion plugin, I commented out the information here that has the field name, the secretion type, these tags over here, and I basically moved them inside the secretion data. And then I'm gonna specify also my boundary conditions to be periodic on the X and Y axis. And always remember, remove the uniform initializer because we're not gonna have any cells. Let me try running it up. Before you run it, let me make sure that I have the right syntax. Okay, so that's not the right syntax because my medium is not secreting. I did upload a copy of this particular exercise as well. This is just as a reference. So if this happens to you as well, this is why we uploaded this information. Okay, so it's just right because we already have the field, so we don't need this field name data here. So we just not need to specify the secretion. Okay, so that should work. I'm gonna stop. I'm gonna relaunch the poll just to make sure that everyone is with me. So as a reminder, I commented out the secretion information in the plugin, and I added this line in the secretion inside the secretion data. And then change the diffusion parameters and my boundary conditions. Okay. Next thing I'm gonna do is that I'll add the plot code that I've shared, that I've been using repeatedly, and I'm just gonna put it in the steppable. So I just have to override my start and step function. So quick question, based on these numbers, what should the equilibrium concentration be based on solving the diffusion equation here? So I'm gonna show the numbers again. So my secretion rate is 0.1, my decay rate is 0.01, and my diffusion coefficient is 0.1. Right. So my equilibrium concentration should be 10 based on how much the medium is secreting, but how much is also decaying. So if I run that simulation, what I should see, so yeah, 1 second. Let's see if it's a matter of, yeah, hold on. Zero 0.1 and it's, I'm sorry, it's 0.0. Okay, now I have the right. Right, so my equilibrium concentration should be 10. Oh, I'm sorry. That's not what I wanted to do. I wanted to share my relaunch the poll. So I want everyone to be with me here. So again, the reason I'm reaching this equilibrium concentration is because my medium is secreting the chemical. And once the medium secretes this chemical, the chemical decays based on the rate that I specified. And then the equilibrium concentration that my field is going to reach is going to be the secretion rate over the decay rate. Okay. So I'm gonna give people a couple of minutes so they can catch up. Remember, all I did was I specified my secretion plugin, but I moved the information out of this secretion plugged in and put it inside my diffusion step hour. And I did specify my global diffusion constant, my global decay constant, my secretion rate, and then my periodic boundary conditions on the X and Y axis. And notice that the equilibrium concentration does not depend on what the diffusion coefficient is. Gonna give people 2 or 3 minutes. Again, if you want to get if you want to have this code as a reference, the name is exercise 242. Okay. Okay, let's continue with the next example. So now what we will do is we will introduce a single cell, and then we will explore what the different secretion options are, and how the concentration profile changes depending on the options that I choose. So what I'll do is I'll build this full simulation with all the possible options, and then we will explore each option independently. Okay, so I'll start by creating, again, a new simulation. I'm gonna call it single source. And I'm changing it to 100 by 100. And in this particular case, the cell is also going to be frozen, so I do not care. And the reason I'm doing all of this with frozen cells is because then the concentration profile is static, and we can actually actually focus on the effect of the interaction between cells and fields rather than also taking into consideration the cell movement. So I'm just gonna call my cell type cell and make sure that it's frozen. Oh, sorry. So how to plot again? I'm sorry. I didn't see those comments. So I'm basically using this default plotting code that I'm pasting in the chat. But again, to generate this plot, I use the pull down menu options and the options on scientific plots. I set up the window and the data series by choosing this option on the start function. And that's how I created these two lines. And then I added data points to the plot by using the pull down menu, scientific plots, add data points inside the step function. And that's how I get this line. And then I measured the concentration of the field by getting this field reference from my pull down menu, chemical field manipulation, and I needed two things. I needed the reference, which is a way for my Python to reference the field that I create, that it's specified in the XML, and then the field value. And this is what this line and these lines are. And what I'm doing is I'm iterating on my X domain to get the value of the field at each position in the X domain, and in the Y domain, I'm just measuring right through the middle, and then I plot it. That's what I'm doing. Okay, so now I can come back to the simulation that I was working on. I was creating a new simulation. I was calling it single source is 100 by 100. I added a cell type and I froze it. I am also adding a chemical field again, calling it oxygen. And then I have to enable the secretion plugin. And I'm not gonna specify anything here because I'll do everything in the directly on the XML or in the Python file. Okay, let me close all of these windows. So in the XML, we have the two cell types, the medium and the cell. And then the first thing is that we're gonna create a frozen cell in the middle of the simulation domain. In this example, I use the blob initializer instead of the uniform initializer if I want to change the steppables once I have created the simulation, I don't have to start over. I can just, let's say, delete this, the uniform initializer steppable, and I can come to CC3D ML steppables and call the blob initializer. And that's gonna paste the code that I need. And then to create one cell, I'm just gonna specify a cell of radius 8 and gap 15, and I'm sorry, width 15, gap 0. And that's gonna create a cell in the center of my simulation domain. Remember that this cell is gonna be frozen. And now there is two types of secretions that I can specify. Well, there's more, but for a cell that is just one cell, let's set my boundary to periodic on the X and Y axis. And let's set my global diffusion decay coefficient to be 0.01 and the same inside the cell. And as I was saying, there is two types of secretion that I can specify in the XML. I can specify secretion inside the cell, which is the one where every pixel of the cell is gonna secrete, or I can specify it as a constant concentration, in which case each pixel of the cell is always gonna have a constant value. And this is equivalent to the fixed and fixed value and fixed flux boundary conditions that we saw, but instead of being applied at the edge of the simulation domain, they will be applied inside the cell. So the first thing I'm gonna do is I'm gonna test the secretion inside cell. Again, I do want the plotting code. So all I'm doing is basically, because I don't wanna have to redo it for every simulation, I'm basically copying and pasting my plotting code and replacing my start and step function with that plotting code. Okay. And now let's execute that simulation. Single source. And I have my cell in the middle of the simulation domain, my measure of the field, and I can see the cell secreting. I'm gonna turn off my contours so we can focus on the colors, because this is important. Okay, I'm gonna launch the poll to make sure that everyone's with me. And in this particular case, yeah, this is secreting inside the cell. So I'm gonna show the code again. Yes, absolutely. That's what I was about to show. So I did specify my diffusion parameters, the diffusion coefficient and the decay coefficient. I specified the secretion rate, and then I specified periodic boundary conditions on X and Y axis. And then for initializing my cell, I just specify a radius of 8 and a width of 15 using the blob initializer. Okay, I'm gonna give people a little bit of time. But now you can see that the cell is the one actually secreting the field. If I wanted, instead of this secretion option, I also have another secretion option where I specified the cell as having a constant concentration instead of a constant secreting rate, which is equivalent again to the constant value flux that I showed. I'm also gonna remove my fixed values. So you can see now that the concentration inside the cell is always a particular value, 1, which is the one that I specified. And I don't know why this is. And then outside of the boundaries of the cell, then the chemical decays exponentially, which is expected, right? Because the cell is acting just as the boundary was acting before as the source of the chemical. So it's important to notice that the shapes here are very different, and those shapes the shape of those profiles might be very important, especially when you're in the proximity of the cell. Okay, I'm gonna continue going to show how to specify the secretion inside cells in Python. So now in this same file, what I'll do is I'm gonna comment out all the secretion options in my secretion data. And then I'm gonna come to my Python step out, and this part is important. And in the step function, again, my best friend is always gonna be the CC3D Python pull down menu. And I have a secretion uptake option. And this secretion uptake function has all of the options that I want for the different secretion options that I have to the ones that I explained at the boundary or inside the cell or outside the cell or at the center of mass. But I'm just gonna show what it looks like when you put one, because all of them are the same. So when you click on it, you get this code that you have to fill in, this code snippet that you have to fill in. You have a secretor object that you need to adapt by referencing the name of the field as is in as specified in the XML. In my case is oxygen. And then I make my cell secrete by calling this secretor.secrete inside cell method. But see that this is referencing a cell. So to be able to use this, I need to specify what cell I want the secrete inside cell function to apply. So the way I can, one of the many ways I can do it is to iterate through my list of cells, which in this case is just one cell. And then here I specify what the secretion rate is inside the cell. So again, CC3D Python, secretion, and I choose a particular option of interest. And I had to specify the field name. And I had to specify what cell this applied to by wrapping the secrete inside cell inside of a for loop. And when I execute it, now secretion is being specified in Python rather than the XML. I'm not gonna type all of the different options because I already did that. If you open exercise 243, and you go to the Python steppable option, you can see all of the options being specified there, as well as in the XML. Right, so in this particular case is specified in the XML. For if you wanna, as a homework, I'll let you play with each of those different secretion types just by commenting in and out whichever of the secretion options is the one that you're interested in having a look at. So these are the ones specified on the XML. These are the ones specified in Python, and see that all of them produce a different type of profile. So I highly recommend, because this one would require a lot of legwork to implement each of the secreting options. I would recommend having a look at this particular example that I shared with you. Okay, so I want to cover uptake. At least the uptake is implemented, basically, it's pretty much implemented in the same way as secretion. The mathematics are a little bit different, so I'm gonna go over a couple of minutes so I can actually explain uptake because it's also important. But in terms of the implementation, I'll just show the code because it's not different from the code that I'm showing here. It's just that instead of secretion, it does the uptake. So cells do take up, of course, they do take up chemicals from their environment, biological molecules, and this is important for respiration. This is also, we can also think of this as, for example, when cells get infected by viruses, they do take up viruses from the environment. And there is different mechanisms by which cells take up oxygen, take up molecules from the environment. But from a modeling perspective, from CompuCell perspective, we can summarize or we can simplify those by thinking that the net effect of uptake is to reduce the concentration of the chemical in the domain of the cell. So it's important to notice that the uptake has to be proportional to the concentration, because of course, how much the cell, if there is no chemical outside of the cell, there's no chemical the cell can take up. So there always has to be, in terms of the mathematical model, we always have to do the uptake a function of how much of it is available in the outside. And we also know that there is a limit, there is a speed limit at which cells can take, or at which cells can uptake from the environment. They cannot just take infinitely fast. So we have to taking those two factors into consideration. The best way to model uptake is by having a saturated function where uptake is 0, is the concentration is 0, and it has a maximum rate when the concentration is high. The way CompuCell implements this uptake is that we have to specify two parameters. We specify what the uptake rate is after a particular concentration, and below that particular concentration, what fraction of that amount it takes. So basically, it acts as a uniform absorption or sink after the concentration increases above a particular average. It takes all of it, and below that concentration, it takes proportional to the concentration that is available. And that is the way that CompuCell approximates the saturating function that needs to be used for the uptake. So when we, the semantics or the syntax for the uptakes are the same. So uptake in CompuCell cannot be specified in XML, it has to be specified in Python. And the uptake is relatively similar to the secretion, except that now we have to specify two arguments instead of one. We have to specify what is the maximum uptake rate and what is the relative uptake, or how much of the or the relative uptake rate that basically indicates the fraction that it takes below when the concentration is below a certain limit. And if we want to see the implementation of this in CompuCell, we can open, because I'm not going to build the simulation from scratch, we can open exercise 244. And again, uptake is not, at least in CompuCell, uptake is not specified on the XML, it's always specified on the Python. And the options are here. Again, these options we can have access to by going to CC3D Python, secretion, and then we have the different uptake functions inside the cell, inside cell at boundary, inside cell at center of mass, and outside cell at boundary. And I'm gonna leave it as an exercise for you to run it. But it does create different profiles, concentration profiles, depending on what uptake form you adopt. And in this particular case, I had the source of the chemical field to be from the boundary. So I set a fixed value at the boundary, but I could also do it by having the medium secrete as we did before. And the profile, the concentration profiles look very different. And this matters in terms of whether the source of the concentration or the chemical is far away from the cell, or the source of the chemical is right at the membrane or the boundary of the cell. Okay, I'm gonna stop here. I'm gonna leave this particular presentation to this particular simulation to for you as a homework to explore. But remember that then there is these two options where you can have either the medium secrete or you can have the boundary being the source, and then all the different options as to how the cell is secreting. And I'm gonna let James run the end of module poll now. And if you have questions, please feel free to ask them now.
I have a question regarding choosing which solver to use. Imagine that we are choosing one solver. So how should we know that the solver doesn't work correctly, or in terms of stability, or we should change the solver? What is the indication of the incorrect solver that we should change it?
Right. So for this particular, so in this particular type of simulations that the chemical field was not interacting except with the cells, and the diffusion coefficient was relatively small, the diffusion solver is fine. The diffusion solver FE is fine. If you have very high diffusion high coefficients, we recommend using the steady state diffusion solver instead, because in that particular case, the molecule diffuses so fast that it reaches the equilibrium. We assume that it reaches equilibrium really fast, so we don't care about the transients. And there is the diffusion reaction diffusion solver that allows you not just to specify the diffusion of the molecule, but also whether it reacts with, for example, with a different field. Let's say you have an oxygen field and you have, let's say, a glucose field, and you want those to interact somehow between each other. Then you can use the reaction diffusion solver such that the fields will interact between each other. But that's going to be dependent on the particular problem of interest. So yeah, there's of course no a priori best diffusion solver to use, except based on the particular problem that you're interested in. AJ, you have a question?
In the wizard panel, there was a choice for enable multiple calls for PDE solvers. What was that? When we were choosing the diffusion solver.
Right. I think TJ can explain that a bit better.
Okay, I can jump in. So the diffusion method we use is numerically unstable for large diffusion constants. And the original version of the diffusion solver, you had to explicitly tell it if you were going to use a large diffusion constant, and then it would know that it should actually call the diffusion solver multiple times per step. That's now done automatically. And so you don't actually need to use that button for your diffusion constant. It winds up that it's possible that if you set the decay constant to be very large, you can get numerical instabilities, and then you have to play with things a little bit. CompuCell does allow some games where, in general, this concept of a Monte Carlo step is the smallest time step that steppables are called in. It's possible to actually substep them models if you need to. I wouldn't worry about those kind of things until you need them. There are going to be a lot of options within CompuCell that we're not going to mention, typically because you don't need them. And so if you leave them set the way they are, you should be okay. But it's true that there are a lot of options. And so by all means ask. In this case, that's essentially a legacy option because we wanted to be able to allow old code to continue to run, but you shouldn't have to click that box. So let me just go back and wrap up quickly. Hold on a second. So thank you, everybody, for sticking it out today. I know it's a long week, and we aren't even halfway through the week yet. So please do answer our questionnaire. Tomorrow we have scheduled, sorry, this is actually, I think I opened the wrong, no, this is correct, but it's highlighted wrong. Tomorrow, we're going to talk about cell properties and behaviors, and in particular, cell division. We're going to have an avascular tumor simulation where the cells absorb nutrient or oxygen that is produced by the stromal tissue and grow in response to that, become quiescent and necrotic in response to that. And then we're going to have a simulation, which Roberto Tomas will show us, which uses chemotaxis effectively to generate 2D and 3D simulations of cell migration. And that'll introduce the idea of compartmental cells. And then in the afternoon, I'll start with a gentle introduction to using tellurium and antimony inside of CompuCell to build network models so that you can control the cells with signaling and regulatory networks. So those were really the things that we had on the agenda. Tomorrow, the schedule will be the same. I'm sorry, I'm not sure why this is showing today's schedule rather than tomorrow's schedule, but it looks the same, except the 2 should be replaced with 3. We'll start about 10 a.m. as usual with questions and comments, an outline of what we're going to do. Then we're going to do when we come back to the previous one, which will show it correctly, we'll come back to the cell properties and behaviors, which Josh will do, the avascular tumor simulation, the 2 and 3D cell migration, and then the introduction to subcellular modeling. And so that's the schedule for tomorrow. If anybody has any specific questions, wants to talk about the hackathon projects, appreciated all the neat suggestions for hackathon. That will be great. There's a question, will Josh please upload the demo files for tomorrow? It would be ideal to be able to upload the CC3D files to NanoHub before the session. That's a good suggestion. I've already put the demo file for 3.4 up. So that is loadable from the student materials folder. There's a question, is it possible to preferentially localize chemicals to different parts of a cell? That's a complicated question because if the chemicals are represented as fields, then you would have to have secretion and absorption in order for that to happen. Julio has developed a method where you create what are called subcells, which represent chemical species and that move within cells, and that allows you then to implement actually the movement of chemicals within cells. But that's a relatively complicated thing to do. I actually showed those simulations yesterday, some of those simulations yesterday. I don't know if Julio is going to be able to get to that within the limits of the course, but if somebody wanted to learn about how to do that, you should ask Julio, and he could perhaps show you how to do it. Yes, there's a comment about using modeling phase separation of chemicals within the cell. That's exactly right. Julio wrote simulations, for example, of PIP2, PIP3 segregation on the cell membrane, of the localization of flamingo and Van Gogh in planar polarity development in Drosophila. using this methodology. And it's fairly straightforward to do it, but it's probably a little bit beyond what we're going to get to in the classes that we have here. If you would like to see that, we can ask Julio and see if he can prepare a module on it, but that's probably a little bit beyond what we can do in this set of classes. But we definitely can do that. There's a question, can C3D solve advection diffusion problems? The fundamental answer to that is no. Solving advection diffusion equation is an industry. Typically, a good advection diffusion solver is 150 to 200,000 lines of C++. And there's specialized packages like Phoenix that do that. There also are some finite element packages or finite volume packages that work in Python that you can use. And so if you really want to do advection diffusion in the classical form, which would be solving, for example, full Navier-Stokes equation, then you're better off using one of the pre-existing packages that are highly optimized. TJ can weigh in on that. If you want to solve advection diffusion using cells, you can do that. You can define a scalar concentration inside of each cell. You can define a chemical exchange between cells, and you can do diffusion that way. There used to be some relatively sophisticated advection diffusion solvers of that kind in CompuCell. I actually don't know, TJ, if those are still implemented or not. They weren't used very much.
Which one, sorry?
The advection diffusion solver where you actually have diffusion happen between cells automatically.
Yeah, they're still in the code base, but I don't think we even document them anymore. Certainly we don't work on them.
So you could try them, but that would be at your own risk. The reason for that, again, is not that's not an interesting problem, but that actually numerically to solve it efficiently is extremely difficult. And it's better to outsource that to packages that are specialized. But it is possible to do it if it's something that you need to do. Any other questions about that? We've certainly done advection diffusion. Actually, I showed you an advection diffusion simulation. The simulation I showed of liver toxicity used an advection diffusion solver. So it's not that it does, it's not possible, but it's numerically not a very efficient way of doing it. TJ's comment is a good one. It's really hard to do it. To develop a general purpose advection diffusion solver is very hard. Doing it for a specific application is not so hard. But to write a general purpose advection diffusion solver that works well under all conditions is quite difficult. Anything else we can talk about? I know it's been a long day for people. I hope that the discussion of the hackathon projects was useful, and I hope that we're able to get some good hackathon teams together. Okay. Well, thank you, everybody. Maybe our instructors could stay on for a second, and we'll look forward to seeing you tomorrow.
Thanks, everyone. Have a nice evening.